نرحب بكم جميعا بالحضور الكرام في النهار الثاني
We would like to welcome you all in the second day of the international annual symposium of the AOAC International, the Arab section. Regarding the first session for today, it's going to be on the food fraud, which is quite an important topic globally and also in the Arab region. We will have a number of speakers to share their experiences in the field of food fraud. We're going to start the first presentation by engineer PMN Rajesh. He is an application manager from Walters Corporation company, the Middle East branch. We would like to welcome engineer Rajesh with us.
Targeted and non-targeted analytical approaches for authenticity testing and identification of food fraud. So, Engineer Rajesh, thank you for your presence with us and the floor.
Good morning to all. Good morning to all. Can you hear me? Yeah. So my name is Rajesh. Actually, I came for some other work yesterday and I was asked to give this presentation in last minute. Maybe it's Dr. Samuel's request. I accepted this. And yeah, I'll just go through some of the requirement in the food industry where food authenticity is going to be very important in future. Okay. It's already, we have started working on that, and I'll be showing you some of the slides which are actually what we can do in case of food authenticity requirement, and how scientists can develop something new by yourself in your labs and create something, your own portfolio and scenario for the countries where you are living. Suppose in the Jordan, you want to make sure that whatever you are importing, you want to see that with a proper authentication and identify what kind of authenticity or biomarkers you can create further your own from your lab. So I'm just trying to flip it. Okay. So when you talk about the good, bad, and fake, these three are very important. Good, of course, if you see vitamins, sugars, whatever you are taking, reading in the food, it is considered as a good ones. When you look at the bad, where the pesticides comes into the picture, and then you have there's allergens, toxins, all those things, you need to be reading them as a bad. The third point, what we are seeing now, is nothing but the fake. So the food authenticity comes into the picture here. You want to know whether it is a meat or it is something else, or sometimes there are many like a honey is one example which I am going to explain today. Though honey is whether it is a fake honey or a true one, or they mix something else. Though that point is very important for us. So when you look at targeted and non-targeted, when you say targeted, you know what you are looking for. That means if you are working for the pesticide analysis, you know that these are the pesticides I am trying to see. And when you look at non-targeted, you don't know what you are searching for. In fact, it could be something, a different one. You want to identify that. And there the challenge comes into the picture. So there are many instruments used in the food authenticity. If you see, you can see the lot of things. In the chromatography, you have HPLCs, GCs, SFCs. Then you have mass spectrometry. You can also have some instruments which are direct infusion. There are SAPs, then we have DARTs. Then there is, again, if you come to the next level, like you have HPLC with MS, HPLC with a TOF system, UPLC with a TOF system. There are many systems comes into the picture. Even NMR comes into the picture. So there are many instruments used for the food authenticity. But it is not a simple job. I can say it's a very complex. You want to see, you are not actually targeting something. You are targeting, yeah? It's not clear? Okay. Actually, if I'm turning, it is losing. I can see that. Where? Okay. Okay. You mean to say I can see the screen? Okay. Yeah, that's better, I think. I don't need to wear the specs. That's important. Okay. When I look at the complexity of doing this such kind of non-targeted screening activity, it is not so easy. It requires a lot of things and a lot of effort in doing those activity. If you see here, you are trying to see something unknown, which is here it is very clearly visible. And among the white flowers, you can see that one red flower. It is so easy. But when you have a mix of all of them, you cannot identify the red flower which is hidden inside the white flowers. So for that, for doing this kind of activity, you required a lot of instrumentation and knowledge behind it and acquisition, and there are many softwares behind it to work for unknown screening. So if you see here, you can see like time of flight machines, which are floor models and also benchtop models. You can find them, which are useful in identifying or searching for the unknowns. So when you look at this, you need to have mass accuracy data. At the same time, you require to do the quantification of the same. So identification and quantification, that's what we are going to deal today. So now come to the time of flight machine, what it does actually. When you look at the time of flight machine and you see this, when the ions are pushed for a longer distance, what happens? These ions can be easily distinguished by their mass by charge ratio. So this works on a principle of moving the ions for a longer distance. What happens? Small molecule can travel very fast and large molecule can travel little bit late. Suppose an example I can say, if you have a ping pong ball in your hand, you just tap it with the 2 fingers, it can just go to the corner. If you have a basketball in your hand, you need to use both the hands to. If I am allowing to push the same energy with the same, all the molecules, what happens? The small molecule can transfer very fast and the large molecule will follow that. By the way, when you transfer for a longer distance, what happens? They will be separated by their mass by charge ratio. Till now in the mass spectrometry, we know only the nominal mass and understanding the nominal mass of the compound. But when you resolve them further, you can see them in a different way. So what is the meaning of resolution here? If you take any beam of the mass spectrometry, you can take the full width of max. That will be the resolution of that particular mass of your interest. What the TOF systems can do, it can resolve them very clearly by their mass by charge ratio so that you can differentiate the elemental composition of the compound. So if you see the nominal mass and the exact mass, this is what the difference you will find. So the nominal mass, if you say 500 the mass of the compound, you can see same number of mass, same mass, 500 number mass. You can see many compounds in this world, but they're all not same. They have their different elemental composition and they are differentiated by the structure. When you look at the elemental composition of a system, elements, what do you see in the periodic table? If you say hydrogen, you say 1, right? It is not actually 1. It is 1 point something else. So if you have seeing this analysis by the decimal place, by exact mass, you can identify the compound of your interest. Like this, you can see here, the carbon monoxide, nitrogen, and ethane. You have 3 different molecules having the mass of 28, but their elemental composition, if you see exactly, they are differed by their decimal places. You can see that the decimal places, you are differentiate them very clearly. So that means this is having different elements, this is having different elements, and this is also having different elements. This kind of identification, you need to know that mass of the compound and accuracy. By the way you're doing what exactly, you are trying to search unknown compounds in your food. So this is how the structure of the system, how it looks like. And here the ions travel a longer distance in the tube, and that's the reason you will be able to separate the mass by charge ratios. Now come to the sample, like a honey sample. So honey is like, if you want to make sure that the honey is geographical region to region, it differentiate. Like an example, if you say you have Jordanian honey, suppose, how to differentiate from the honey from the different country, which comes from the Saudi, or it comes from the different parts of the world, maybe Australia, from New Zealand. So how to differentiate that? And by the way, and also here the point is food fraud. Suppose if there is something like a good kind of honey, it charge you more, right? There are different classification in honey also. Some of the honeys, they are very high cost because they are from the particular region or particular source. So if I add something mixed in that, nobody knows it. Both are sweet, right? So obviously, what you can see, how I can say that there is some adulteration in that, or how I can authenticate that, whether this is present or absent. So this is where the mass spectrometry instruments comes into the picture, and why I am talking about that exact mass, because you are trying to identify the unknown, untargeted compounds. So if I take food security, we need to see that every food which comes from the outside of a country, there is a quite chance of mixing of some adulteration can happen, mixing of the meat.
يعني يمكن خلط اللحوم بشيء آخر. وقد سمعنا أن هنالك أيضا عملية خلط للقمح. يعني عندما نشتري السلع الغذائية فلا نعلم بما هي مخلوطة، وعندما نتناول طعام نحن لسنا متأكدين 100% إذا ما كان تم مزجور شيء آخر. حتى ماكدونالدز يعني هو إذا ما تناولت الدجاج.
That how I can authenticate that. So these are all the things are very important. The same way the honey, what we have done, I'll just try to show you that. So if you look at the products, every product which comes from the source, by the time it comes to the consumer, it is definitely get adulterated. In India, milk is adulterated. Some other place, some particular food, source of food will be adulterated, because which is in high demand, that can be adulterated very easily. And we have to know that before we understand that whether it is adulterated or not. This is the path I am showing you here. So from where the source and by the time you reach to the consumer, so there are many if and buts. You can see here. So anywhere, any place, the adulteration can be possible. So you need to keep an eye on every place of, the source, from the source to the consumer place. So you can see that some of the chocolates are banned in 55 countries because we don't know what they are actually mixing. This This is one of the reason why we get into the picture of adulteration. Now, when you go to the honey, so honey got a lot of things in that. If you see here, they have flavonoids, phenolic compounds, then you have sugars, natural products, and we can also have a fingerprint depending on the region and the biological activity and also the force targets. This you need to see what kind of things we need to analyze and try to understand from the honey. Here, one example, what we did is in the lab, it's not working. Yeah. So if you look at here, this is what I was talking about. This Manuka honey is costing 55 pounds. So this is a very high cost with a little volume. And also it was, why it is so costly? It is also used in the treatment. So some of the cases, it is treated as an anti-inflammatory. So now only I need to know whether it is Manuka or it is something else. And if you see here, we have done a lot of work on this, and we have taken the different kinds of honeys from the different regions. If you see here, we collected everything from the different places, and we did we have given the importance to the microscopy, first one, but microscopy will not give the differentiation in the pollen grain. Then we try to see that NMR can produce something good, but still we could not come to the conclusion that whether it is differ from the other honey. And when we go to the mass spectrometry instrument, we have found something differently. This is the NMR results, what we have found. But it is not going to differentiate. The potential biomarkers are not differentiated from the one honey to the another honey. But when you go to the next level, that in the mass spectrometry instrument, what we have done, we have taken a complete procedure what to be adapted on that. Look at this. We made the selection of different places. We collected it. And we have now the different groups are here from the different countries and different places. But they are same honey here in case, but it is collected from the different places. Here also, we collected from different places. And we have, again, different places. So we also collect the Manuka from New Zealand. So you can see maybe you can see that there are 3 or 4 different honeys, but they are collected from the different regions. Now, when you go for the analysis, what we did, it is very simple. Like we have taken the honey and just you add the formic acid and just, precipitate the proteins by just using simple procedure like a methanol. And then we extracted that and we just centrifuge it. Okay, sonicate and centrifuge and then collect the sample. And we follow the mobile phase conditions with a gradient running with this kind of buffer. And we have the gradient here. Now, after that, and ESI negative mode, we did the data acquisition, and you can see that. So when you make the data acquisition, we see that repeatability of the data from the one group to the another group, and then we compare the data. That's what we did it. So when you make the comparison of the data, And the ProGenesis software will actually help you here in doing that. In the case of ProGenesis, what it does, it has a linear dynamics processing, and it takes any data, any mass spec data, and it makes the comparison and see that who are the new things. Like it is a process. I think you might have heard about principal component analysis. So where we take the data into consideration and compare the data and see that who are the new things. So you need not do anything, and the software will do everything, and it will just make the comparison and find out what is new. So here you can see the procedure. There are two things here. For proteomics, we have ProGenesis QI and we have ProGenesis QIP. QI for the small molecules, QIP for the large molecules. And the protocol is very simple, and you just acquire the data and transfer the data into the software. So what it does the procedure like this. It aligns peak detection, and it makes a deconvolution or a quantification, identification, and then it goes to the statistics and it goes to the pathway. Now, what we are, so after doing this, so the data looks like this. The pool data looks like this. You can see here, we have now here 1, 2, 3, 4, 5 different groups. There is a heather, manuka, buckwheat, pool, and rape. So all these different group of honeys are collected and injected into the mass spectrometry, and then we can see that differentiation. And it is the scope plot where you can see the differentiation of the molecules and differentiated in the region. And then it's a 3D picture. You can see that how they are differentiated in the space. And when you go to the S plot, here the actual difference can be identified. If you look at this place, these are the two different groups, and they are differentiated by the x-axis. If you see here, these are away from your y-axis. Those compounds can be just identified very easily by the software. So by identifying like this, you are going to try to see the biomarkers of that particular analysis or biomarkers of that particular compounds. So same way you can do it in your lab and just do that kind of, if you are doing the differentiation between the food which is imported from the different countries and compare with your country and see that what are the biomarkers of that particular analysis, and you can store in your lab and store in your library, use that as a authentication. And after that, it is a very simplified analysis by using the tandem quadrupole or single quadrupole detector also is quite possible. So once you identify that from the softwares using these libraries, you can directly go to the quadrupole and try to find out like in this case, in case of manuka honey, it's a leptospirin is the one of the compound should be observed in case of manuka. In case of non-manuka, you will not find that peak. So this is how we did this analysis just to authenticate. That means food authentication is quite important. So I just want to give some, glimpse of that. In fact, maybe I think it's more scientific because you need to work out in a different way. So, but it is quite a good example for you to make sure that you can go through this and try to work out on the same. With that, thank you very much for your patience.
Thank you so much, Engineer Rajesh, for your valuable presentation. And already we can see the importance of chemometrics and data treatment in the detection and quantification of food fraud in all its aspects. And here we saw the aspect of authentication of food. So thank you for this valuable presentation. نرحب أيضا بكل المشاركين معنا عبر تقنية.
We also welcome all the participants joining us via Zoom. You are all welcome. You can always participate, ask questions, or raise your hands to speak. We also would like to remind you that there is simultaneous interpreting provided through the Zoom webinar. You can listen to your preferred language, and as you have noticed, we were supposed to start a program of today with two presentations from two colleagues from the SFDA of the Kingdom of Saudi Arabia, but we would like to apologize on behalf of our colleagues because they are unable to join us due to inevitable circumstances, but we hope that they will be able to join us in future events. One more clarification, we would like you to keep your questions for the Q&A session after the three presentations. Now we would like to welcome our second presenter, Hamza Al-Qadiri. He is a professor in microbiology of food and water from the University of Jordan. You are most welcome, Professor. In his presentation, he will talk about new methods in food and water testing.
Good morning.
Good morning, everyone.
I'd like to thank you all for giving me such opportunity to talk about an important topic, food fraud, and new approaches actually to detect contaminants and chemicals, toxins in the food system. This is an old and new topic. Actually, after September 11, the federal government in the US started to think about non-classical methods for detecting toxins, chemicals, contaminants in drugs or in food. Actually, they start to apply these techniques in airports to make sure that there is no bioterrorism. So we start to work with them in 2001. That time I was a PhD student in Washington State University in Seattle. So we start to work in two techniques. The first one, which is called the shortwave near infrared spectroscopy, and mainly to detect spoilage, to detect protein denaturation, lipolysis, whatever. And the second technique, which is the important one, using the FTIR, which is the Fourier transform infrared spectroscopy. And we developed a data bank, a spectral fingerprints for most foodborne pathogens, such as E. coli 01577, Salmonella, Campylobacter, Pseudomonas. After 4 or 5 years, then we started to develop also using the FTIR method to detect enteric viruses in food and in water. Okay. Yep. So, you know, as a little brief, in the food fraud, so to improve the performance of food safety system and to control foodborne diseases. Also to improve performance of food system in the central reaching sustainable development goals, expanding digital opportunity to include farmers, agribusiness, improve food system outcomes in both rural and urban areas, and finally, use digital non-classical technology, which help reduce the cost, time saving, help farmers make more precise decision, and improve access to information, knowledge, and markets. Okay. So we are looking for more resources efficient and climate resilient, such as precision in agriculture, gene editing, biological-based crop protection, technologies that improve traceability, which is very important right now, the traceability from farm to fork. So, you know, actually, we have two techniques, which is the classical, such as detecting the fatty acid profile in olive oil. This is a classical technique to detect food fraud, or the non-classical, non-targeted analysis, such as the fingerprinting, for example, melamine in milk powder, and that's what we will talk today about. Okay. The common adulterated products in olive oil, in fish, organic foods, milk, grains, honey, green tea, coffee, spices, wine, and certain fruit juices. So we are dealing a lot of cases regarding food fraud, mainly in such foods. Now, the 7 principles way as a food or food ingredient can be adulterated to increase perceived value by substitution, unapproved enhancement, concealment, mislabeling, dilution, counterfeiting, and gray market production of theft or diversion. So, as a comparison, yes, here the summary comparison between the targeted, which is the classical method, and non-targeted approaches for detecting food fraud. The most important thing that the classical is targeted compounds, whereas in the non-classical is by fingerprint. So what we need is a database, a fingerprint or data bank. The second thing, the sensitivity. Okay, the classical could be high sensitive. However, the non-classical, it's high throughput. Sample preparation in classical is selective. However, in the non-classical and selective. Data analysis, we can use univariate or calibration. However, in the non-targeted, non-classical method, we can use multivariate modeling, which may require chrometric software. And finally, the targeted or classical methods, very simple. However, non-targeted is complex and it should, need experimental design. Okay. As you see here, this is what we call the principal component analysis. It's mainly used as a separation technique or to make clusters. So as you see, we can distinguish between normal milk. This is the normal milk. This is milk contaminated by melamine, and this milk contaminated by cyanic acid. So this is non-targeted, non-classical fingerprinting technique, and we use the principal component analysis. Now, to understand this, this research actually done in 2002 and published in 2008 in Journal Food Protection. This research done in the US. Sorry. Yeah. No. Yay. This is the FTIR. No. Yep. So here, milk. We made different scenarios for milk spoilage. And we keep milk at different storage conditions, let's say 37, 21, and 6 Celsius. So by this, we enhance spoilage process for milk to detect other, chemicals by, microbial spoilage or by chemical spoilage. This is the raw spectra, what we call the raw spectra. So we use shortwave near infrared between 600 to 1,000, 1,100 nanometer. So as you see, this is the spectra for the milk kept at 6 Celsius. This one for milk kept at 21, 37, and this is the control. Okay. Now, the second one, the same thing, but the difference is the time. I think here, mainly, a 3 hour, 6 hour, and 30 hours. So that's what we call the primary or raw spectra. Actually, we can't use it to distinguish between different kinds of spoiled milk. So we should use the second derivatives. These are the second derivatives for the same spectra. And as you see, we start to detect metabolites or chemicals, whatever, this region. So we have a kind of database to distinguish between different spectra. Till now, still, we don't use the statistical analysis to distinguish between. The most important thing, when we use the principal component analysis, as you see, this is control milk, 0 hour. This is 3 hour, 8 hour, and 30 hour at 6 Celsius. The same thing here, but the temperature is 21 and 37. So we could separate or distinguish between different kind of milks according to spoilage, regarding temperature and storage time within 2 hours. Within 2 hours. So the process doesn't take more than 2 to 3 hours, okay? However, if we would like to use classical techniques to detect microbes, the increase in total aerobic plate count, blah, 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 maybe we need 24 to 48 hours, okay? So this is mainly used, we can't use shortwave near infrared to detect pathogens, to detect viruses, to detect bacteria, no. It's mainly used to detect metabolites, to detect chemicals, to detect, let's say, we could use it for aflatoxins, for example. So, right. Now, another statistical tool, which is called the using the accuracy level to check whether we are accurate or not in separating process. So, as you see, the control is 82% accurate to be distinguished from other treatments. Milk kept at 6 Celsius for 3 hours, 83% accuracy in detection or in separation. However, the milk kept at 37, 90% the accuracy in detecting that milk, or 88% at 8 hours and 92% at 30 hours. Okay. Right. Also here, these are the detection of protein, proteolysis due to spoilage. And as you see, we do have different spectral level according to the storage time and storage temperature. Same thing, we should use statistical tools to distinguish between. This is the second derivative. Also here, actually this experiment done for chicken spoilage. It's not for milk. This is one for chicken spoilage. And as you see, we could distinguish between different samples kept at 6 hours, 0 hours, which is control, 6 hours, 12 hours, and 24 hours without using classical microbial analysis or the total aerobic plate count. We know that beyond 10 to the power 6, 10 to the power 7, the spoilage symptoms start to increase. However, we could detect spoilage even within 3 or 4 hours using shortwave near infrared spectroscopy without any microbiological analysis. Okay. Now, the second approach using FTIR, Fourier transform infrared spectroscopy. By the way, the shortwave near infrared, the cost is about $15,000 to $20,000. This one between $25,000 and $30,000. However, the problem in the database, the bank, you need software, you need fingerprints. So that's why we should start this in Jordan to make our data bank, to make our fingerprinting. So if there is a cooperation between different sectors, governments or private sectors, to develop, to have these pathogens, let's say from different laboratories, whether it's private or governmental, and then to start to build up our database, that would be very excellent approach. So these are the spectra of different pathogens, bacteria in food system. This is the instrument we use, the FTIR. So we are focusing mainly in this region which is between 900 and 1,600 per centimeter. So most of protein, here the protein, here the DNA, the carbohydrates. So we can detect any change in the cell wall of the bacteria in this region, DNA denaturation in this region, and protein denaturation in this region. Okay. So that's what we call spectral patterns for different chemicals. As you see, for example, at 3,500 per centimeter, we can detect the OH bond, 3,200 the NH bond, and whatever. We are focusing in protein, which is the amide 1, amide 2, in the range between 1,500 to 1,680 per centimeter. Also here, the amide 3 in the range of 1,200 to 1,300. Okay. Now, this is the second derivative. So as you see, for example, heat-treated food samples, we can detect the bacteria, the injured bacteria. Sometimes we have a problem, maybe the microbiological people, you know that in injured bacteria or viable but non-culturable bacteria can't be detected in laboratory due to many reasons. So you have to do enrichment, to resuscitation, whatever. So we could detect heat-injured bacteria. We could detect viable but non-culturable bacteria without using any microbiological tools, just within 2-3 hours using the FTIR method. So here, the differentiation, the spectral differences between control, non-heat treated, heat-treated the bacteria, and also this is in the protein region, the amide 2, this is the amide 1, and here we are talking about the DNA denaturation. Same thing, but different wave number between 900 to 1,300 per centimeters. So after that, we should use statistical tools. This is what we call loading plots. Loading plots, actually, as you see, we have PC1, PC2, PC3 also. Now, PC1, let's say 70%. So 70% of distinguishing between different bacterial cells depend on PC1. As you see, PC1, the loading plot is very high, whether in positive or negative. Then PC2, about 11%, PC3, let's say 5%, PC4, 2%. So the most important one is the PC1, mainly 70%. And the most difference is actually in the protein region due to protein denaturation. So we know that heat treatment of bacteria will cause protein denaturation. So as we see, protein denaturation is very distinguished in this region due to heat treatment. Here are the principal component analysis. And as you see, this is the control, non-heat treated. And here are different bacterial cells, depends on in heat treatment. So we can distinguish between also E. coli, between Pseudomonas, between different gram-negative bacteria, different gram-positive bacteria. The most important thing, the clustering. So you see the control is most clustered. That means the treatment's very close together, okay, and separated. Then this is a different treatment, and different treatment. So this one is good. This one is good too. But as you see here, let's say the treatment's far away, you know, far bit. So we try to keep different treatments close together as we can. And this depends on the calibration of the FTIR and other experimental issues. Okay. This is the dendrogram also, using the spectral patterns, using the FTIR methods to distinguish also between different strains of the bacteria, or heat-treated, or using UV light or irradiation, whatever the method is. Okay. I have 2 minutes. Okay. I'm done. So also, this is the principal component analysis. You see, to distinguish between chlorinated E. coli, you know, we chlorinated water, with E. coli and Pseudomonas to see if we can distinguish between chlorinated and non-chlorinated water samples and to detect the chlorinated injured bacteria. Okay, so for example, this is the control, non-chlorinated, and we use 0.3 ppm chlorine, 1 ppm chlorine, and sodium hypochlorite treatment. So according to the principal component analysis, we could distinguish between chlorinated treated, non-chlorinated treated samples, and to distinguish between different bacterial strains, not only between the same strain, but also between Pseudomonas aeruginosa, E. coli, Legionella, whatever. And this is the SIMCA classification system. It's a statistical tool. It's called SIMCA. And it's mainly used to give you an idea about the accuracy or the precision of your classification or the distinguish process. For example, we could separate the control by 93.3% the accuracy. For E. coli, 88.3%, Pseudomonas, the same. For mixed culture also, between the two, also we could make a separation by 83.3% using this technique. Thank you. Actually, I think I don't have more time, right? So thank you very much. And I'm ready for any questions. Thank you. Oh, really? Yeah, okay, thank you.
شكرا جزيلا. Thank you so much, Professor, for your
Thank
valuable.
you very much, Professor, for your valuable presentation.
Again, other examples about the importance of these non-targeted approaches in food fraud detection, the combination between analytical fingerprinting using different analytical methodologies with appropriate chemometric tools in order to extract the needed information. وهنا أرحب بالمتحدث الثالث في جلستنا.
I would like now to welcome the third presenter in our session for food fraud, Dr. Mohammed Hijaz, who will be presenting on food fraud and latest technologies for accredited detection. The floor is yours, Dr. Hijaz. Good morning, everyone. First of all, I would like to extend my thanks to the Arab Army and the Arab section of the AOAC for organizing this symposium and for inviting me today. I'm going to talk about food fraud and the latest technologies for accredited detection. The lecture will be divided into several sections. First of all, we will have an introduction. We usually talk about economic materials that have a high value, and this is where we usually detect most fraud cases. If you look at the screen, you will notice that fraud is at its highest in milk, then olive oil, then honey, then saffron. So we can divide them into different food products with high percentage of fraud. This is an old USP statistics study, but it's still a good indication. Now, fraud usually happens in Arab markets to gain more profit. Recently, the topic of food fraud has become under the highlight increasingly, and we noticed that in Brazil, most olive oil in the market had a high percentage of food fraud. The same applies to Britain, Germany, and as my colleague said earlier, the value of food fraud annually is about 50 billion. I always see an example of something very bizarre. For example, from the very famous Manuka honey, which is known for its benefit, the produced in Australia is about 1,700 tons, but what we see in the market is about 10,000 tons. So this does not make sense.
This time we did not discuss olive oil, but we will cover it later on because we import lots of honey. This is why we're focusing on honey. And it is known that honey is mentioned in the Holy Quran, and everybody likes to use it because of its benefits. Governments are also targeting this topic. So you will see that in the US, for example, new technologies are being applied to detect food fraud. Also, India is paying more attention to food fraud because it is one of the biggest exporters, and they have spent lots of years building a good reputation in food production and export. So what are the latest technologies that we use to detect fraud in high-value products? For example, in honey, usually the fraud methods are either adding sugar or false declaration of origin, or unripe honey, or inappropriate bee feeding, or transshipment, meaning that we ship it from one country, we relabel it, and we sell it in the market, claiming that it originated from another country, and of course the filtration. Now, regarding honey, why would there be false declaration of origin? First of all, it is to hide the country of origin from the customs, because there are usually international agreements stating that if honey is imported from a specific country, then there are higher taxes imposed, or using specific methods to do pollen grains manipulation to deceive detection by pollen analysis, so that this fraud is not detected during this screening. The conventional techniques used now is the two precious mass, such as the HPLS, the LCMS for the markers, and the foreign enzymes. If we want to know what the origin is, we use the microscopy and the foreign analysis. The markers can be removed, the syrups can be filtrated, and the sugar cane can be discovered in another method. Here, as you can see, using microscopy, the limitation is that you need someone who is highly competent with long years of experience to be able to detect it. You need the honey to be unfiltrated, and the expert needs to be able to differentiate between the real pollens and the exogenous ones. Now, what are the types of fraud? Usually we do the sugar profile in codex, but the foreign sugars are sometimes present because there is inappropriate feeding of the bees. Al-Marshud are one of the biggest producers of honey, and they discovered that sugar syrups are present in 10% of the honey available in France. So as a summary, as we said, we should be doing pollen analysis, we should be using microscopy. And if we want to make ISO 2 pressure mass to discover cane sugar, we can also do LCIR mass. Also the sugar spectrum HPLC, SMP, SMR markers, and for the oligosaccharides, the HPLC and the foreign enzymes. When we implement a full profile for the honey, it will cost no less than $1,000 and no less than 3 days. So as representatives of Prober, we are using the NMR to do untargeted screening. Why is the NMR powerful?
بيومين الاس او بيز كلها جاهزه فولي اوتوميتد وما بتحتاج ان ام ار اكسبرت. انت هون الاوبريتور بيقدر يشتغل.
Need an NMR expert. Any operator can use the device. It also reduces the costs, and the report that you get from this solution is accredited as an ISO 17025 because the database used consists of 8,000 well-characterized samples from around the world. Yesterday, we were talking about the importance of centralization and how we can find a standardized method that is accepted in consensus between everyone to be considered a solution. So the food screener can be something used worldwide, and it could consist of uh, of good data. It's also transparent, and we are always ready for roundtables to sit together and to see what we can develop for the future. So this method will be continuously expanded and updated. Now, what is the workflow of the food screener? You, as a user, will receive honey at the laboratory. You will dilute it, and then you will put it in 900 microliter, and then you will have an automated addition of buffer solution. You will put it as 600 in an NMR tube, and then the process will be fully automated. Then the data will be encrypted and transferred, and then you will receive an accredited lab report directly from Bruker. This was applied by Bruker to the most commonly foods where fraud is detected. So we will be adding more and more modules. The available modules currently are honey profiling, olive oil profiling, juice profiling, and wine profiling is ready. And we will be adding spices, including coffee. The latest version that was launched for honey was Honey Profiling 3.0. So what would this lab report present me with? Here, 28,500 authentic samples were done, and there were about 2,000 adulterated samples. The samples were taken from more than 50 countries and more than 100 botanical varieties. So the monofloral and polyfloral honeys from a single country source. And there were blends of honeys from different countries. From the USA, were about 800. From New Zealand, there were 200. And several other countries from around the world. The Arab world is still not covered, but hopefully we will be able to have a partnership with the AOAC Arab section to cover the area. What can the honey profiling 3.0 provide us with? It can detect sugar syrups, as you have seen. The study, which was issued by Femil Michoud, detected only 20% of adulterated samples. However, we managed to detect more samples, and the NMR was the only technique that managed to detect the food fraud. Also, the NMR can use sugar syrups for the verification of labeling of origin in only one run. We can also do detection of atypical samples, which are outside the database that we have.
ليش هي atypical sample
and
بعدين
whether pollen has been added, and at the end,
methods بالregulated parameters على سبيل المثال
there's an analysis of conventional methods and regulated parameters. For example, we can create
هدول بيكونوا
up to 60 parameters per sample of sugar canes, and these values are compared to reference thresholds, and they are all validated to the database available.
أكثر من
And you can manage to detect the adulterated foods from several countries. So quantitatively, I have not covered all the parameters, but based on the codex that we have an absolute quantification for the glucose and fructose, sucrose, and the HMF. And you will receive a report to indicate whether or not the food is compliant or not based on the ISO or codex that you are following.
تمام التارجت اناليسيس برضه احنا حكينا الامينو اسيد دائما بنطلع
As per the targeted analysis, we also look at the amino acids, citric acids. All of these will be covered.
detection of the false declaration of origin انت المفروض تعطي
As per the detection of false declaration of origin, the honey profiler should be informed about the country of origin. So you could tell them that this honey, for example, is imported from Jordan. So when they do the international comparison, they will tell you that the sample you have matches India. So how come it is declared to be from Jordan? So you will have a cross-validation of more than 98% and confirming that it came from another country. This is one of the examples that of the sections that you will see in the report. You will also be able to know the complete chemical composition of the honey, and you will also receive the probability of the sample if there is any suspicion. As per the atypical samples, what they do is statistical comparison with normal profiles, and you will see here, for example, the multivariant verification, and you will see, for example, here the result, no deviation has been detected. So in summary, we can say that there are several international methods, but the idea is that we are trying to reach a method that is easy, accredited, and that gives accurate and reliable results. We said yesterday that accuracy is very important in the probability of your sample. So here as you can see, there's an overview of the testing. You can see that there are the setup markers, the honey, the foreign enzymes. When it is very low, it's like 25% detective. You will notice that in the NMR, we can detect up to 70%. We still have not reached 100% of adulterations, but following conventional methods, the percentage is much less when it comes to syrups. As a summary, the food screener is a push-button solution from Bruker. You will have the SOPs ready and the tubes, and you simply need to apply to it. It also relies on a huge database amounting to 18,000 samples. And now we've reached about 28,000 well-characterized samples. Also, you don't need NMR expertise, and it's very fast. If you compare all the parameters that you will receive from the NMR to the conventional methods, you will discover that you will need 2 to 3 days of work for the conventional methods, and it will cost you about $1,000 compared to the NMR, where you can process the sample within 20 to 25 minutes. Also, it is very difficult to deceive the food screener. There's always gonna be a new way that fraudulence are trying to deceive, but it is very difficult to deceive. We are always looking for a wide and worldwide network of partners to expand our work. Currently, most commonly users of food screeners are in Czech, Australia, Germany, Spain, India. All of these governments have the food screener NMR, and these are the agencies in these countries or private companies that own the NMR because they have large amounts of exports. Thank you very much for listening. Thank you very much, Dr. Hamza, and I now invite Dr. Qadiri and Mr. Khalid to join us.
If you have any questions, please do not hesitate, and then we will try to have a brief discussion. اتفضل دكتور.
صباح الخير جميعا مرة ثانية.
صباح النور.
Good morning, everybody. I believe 3 lectures, in my opinion, will change the world with the fraud, with the analytical test, and also I also blame the brokers, and I will tell you why. Of course, Doctor, I would like to congratulate you. You've been working on the project since 2003. And frankly speaking, the laboratories of the Northern Governorate, we've been working on it for 7 years. I was quite happy for the Arab section of in the AOAC International for continuous networking. So you mentioned about water company. You talked about some techniques for alteration. So what parameters can be chosen or selected?
So basically, once you are using QTOF system, so it is an independent analysis. So what we are doing is that we are fragmenting the molecule on high energy and low energy.
Yes.
Take a complete spectrum. Okay. Take a complete spectrum, and then after that, collect the spectrum for principal component analysis, identify the markers, and then upload this data for the Matlin so you can identify the compound. So it's an independent approach, non-biased approach, in which you will fragment the molecule and get all the fragments, compare with the library. If it is present, that is good. If it is not present, then go to the Matlin, identify the markers from that one, and then publish it.
Okay, thank you. Mr. محمد أو دكتور محمد، نحن بصراحة broker wise.
Mr. Mohammed, broker wise, we have been working with you for many years, FTRI, Wango, and liquid olive oil, and milk, and XIR. So what you have said regarding honey in particular, I don't share your opinion that this is going to be the master. I will tell you why. I'm not saying this because too many people are committing a fraud with honey. We have information from the open source. We have a specialized person who is specialized in the matrix, and we conduct coding in collaboration with the laboratories. And we have a protein lab where we collect the data for the collected samples, and we are doing our own library. We have created our own library. Like Agilent has its own library and the rest of the known-renowned companies. So my direct question for you, I hope, like you said, that the NAR is available in NMR is available in all the world except for the Arab region. In Germany, does it cost $1 million? So we would like to invite the AOAC Arab section to collaborate to have it for 10 years. We will share all our data of honey regarding the spirology, regarding the pollen test, and Melissa pollology. It's been since 2 years in Lebanon, we have 2,700 flower species. We are now creating our database. It's Atlas database for Lebanon. And I believe that Jordan is similar to Lebanon in nature, the same for Palestine and Syria. So we created the database that includes the type of flower and all of the other characteristics. So I invite you seriously to think about this idea, since the Arab world is part of the global world, and we all suffer of the same issues. Thank you very much. Another point you have commented on, the method is not accredited as 17025. However, it's validated but not accredited because you cannot conduct an assessment. My question is, the machine can be an assessor for 17025? Is it accredited by the ISO 17025? Because all the FTR and the other similar products we suffer from. So the person who commits the food fraud would argue that this device is not accredited, and they will argue that they did not commit any kind of fraud. Broker realizes this issue, and we are working on the accreditation. So your report is issued using the NMR technique is considered an accredited report if you follow the SOPs. So in broker's lab, you would find in many cases we use the report resulted from NMR and was considered as an accredited report in the case. So my last question, to Dr. Qadiri, regarding FTNR, the work is at the microbiological level, and it's quite important. As you know, the Salmonella, Listeria pre-screening or testing, is important for the pre-screening. Especially if there is a flash pandemic in the country, you want to have expedited results. For instance, if there is mass food poisoning from that resulted from a certain restaurant, you have to conduct a speedy test for Salmonella. And Listeria, Salmonella plus positive, and E. coli H7. These are the 4 most hazardous food pathogens. Maybe you can help us to benefit. So do you represent a university or a company, and do you work at a research level or a laboratory level? Because currently I'm networking our laboratories with you. So and I would like to take permission from Dr. Biyazbrek to talk about this, because we are establishing a center in collaboration with the Ministry of Economy to combat food fraud at the national level. This is a collaborative effort. In Lebanon, we realized that we are part and in the middle of the Arab world, and the Arab world is in the middle of the world. And food frauders have a high level of thinking that is frightening. And as for honey, 6 months ago, there was a honey exhibition in Beirut, in Lebanon. Thank God, the cooperatives and societies, the farmer societies and cooperatives are quite aware of all the information related to the honey industry, and they provide the most popular and rare honey products, and we received a number of complaints that the number of complaints that there were a type of honey sold very cheap, and they suspected there is food fraud. After testing, we found that the glucose syrup and pollen, a sprinkle of pollen, were used in the honey fraud. We did 14 tests in order to confirm that this type of honey was not original. After 2001, the research-wise, our work is a research-wise between 3 universities, Idaho, University of Washington, and the University in Seattle. We are supported by the US FDA. The database is based in the United States. No one can access except for the persons who are working on this topic. The German had worked on it since 1998. Scientist Newman is still alive. He is the founder of this topic, and we continued. The device was used in 3 airports in the US, 2006, 2007, and 2008. And currently, they stopped these devices because the bioterrorism had been reduced. The work is still, the research is still ongoing, and the application does not exist outside the boundaries or the borders of the United States. However, regarding networking, since 2007, they started to work on PulseNet system. The PulseNet system connects all the states in the United States through the CDC, which is the NDA fingerprinting, and it's used for different pathogens, and they can see what is the source, what is the bacteria, or any other new things using the Pulse Nexus system, which is based on the next field, and it's accredited and in use and in place in the United States in collaboration with the CDC. However, what I want to stress is that our work is research-wise, and we can't, here we're talking about exotoxins and whatever. So this is research-wise is in place. So the problem with research is that you need funding, and you need dedicated people to allocate their time and efforts, and you have to realize the database is inaccessible unless you are authorized. Even I cannot, do not have access. Why not establishing or creating our own database? When I came back from the United States in 2005 and 2006, I made the suggestion. However, no, there was no response. Doctor, if you would allow me, we have started with the database. We found, starting from our lab in collaboration with the Chamber of Trade. Dr. Jadriz Kalla is my colleague, and he is a specialist who has 2 patents. He used to work in France with the most important partners in the FDA. However, maybe during the break, we can discuss this in order to build a database that can be used not only by Lebanon, but the Arab countries as well. As for the diseases, in Jordan, do you have Do you have Castrol? I'm talking about the classical. For a certain outbreak, right? Do you have isolation for a certain type of outbreak? This need to be requested, she answered. The CDC in Jordan has a hand. I believe the Ministry of Health can answer this question. I believe if we have an isolation and then do screening and then will take the fingerprint, this is part of our work. We are the Lebanese university we have bacterial bank that is 30 years old. We have all the germs, diseases that that spread or outbreak in Lebanon. We split and we can multi-TOF screening. So we are ready. We are already ready. We can start 4 isolations and sometimes we do isolation for cheese where we find salmonella or other diseases, and we can collaborate to work on other products as well. Thank you very much. We still have some time for one more question. Can you please speak through the microphone? My question to Dr. Mohammed. I'm from Hijazi company. Regarding the food screener, regarding the honey testing, what about the other modules like olive oil? For olive oil, we have 2.0 that is available. It provides complete results. It provides 15 AOAC parameters that are accredited. We conduct target screening also on olive oil, and the module is ready. And also we have module residues, which is ready and complete. I did not mention it in the presentation for time constraints. And also for pomegranate. The modules are ready. We have module while for spices. It's not issued yet, but it will help the copy as well. It will help saffron for anything that food fraud is high. It can be to which presenters and panelists, and as a targeted products, especially the local products that are produced in the Arab countries. We need to launch this type of work in order to expand the fingerprints from the analytical methodologies and techniques that can cover all the physical and chemical and microbiological characteristics of these products in the Arab region. We would like to thank your presence here with us, and now we will take a break until 11 a.m. After that, we will reconvene. Thank you very much.
Hello, everyone. We will now reconvene and continue with the next session, which is dedicated to pathogens and gluten in foods. I would just like to remind you that simultaneous interpretation is available, and you can reach it through Zoom. And I believe that there are several presentations which will be in English. I will also be using English to facilitate the work. So please listen to interpretation.
Privilege to be chairing this session. We will hear a number of presentations dedicated to the introduction of food allergen and gluten management. We will also be a number of analytical tools for the measure of gluten sources and also the determination of allergens in food. And we will have participants from Jordan, Lebanon, as well as our partners, our BioPharm from Germany. I will be starting the first presentation, and this presentation actually is meant to offer an introduction to food allergen and gluten management. So what I will attempt to do during this presentation is to review the various approaches for food allergen management and how food allergen and gluten management actually is included in the context of food safety management systems, and the way to the importance of testing methodologies for the identification of both gluten markers as well as food allergen markers. Now, the issue of food allergy is not a new issue. I'm sure that many of you have at least one, if not more, members of your families suffering from food allergies or disease. Now, the issue with allergies is that the hazard is a foreign agent. It is, in fact, a component of the food. It is an ingredient. So this ingredient is safe to the majority of individuals, except that there are minorities who may be developing a reaction if they are exposed to this ingredient. This reaction is either a reaction that is IgE mediated, so essentially immunoglobulin E mediated, and in this case it would be an allergy, so it's a hypersensitivity of a allergy. And in this case, symptoms from simple itching or urticaria all the way to anaphylaxis and potentially the death of the individual. So the repercussions can be considerable for a food allergy incident. Related to action to an ingredient, they are mediated via IgGs or potentially IgAs. And in this case, essentially what we call celiac disease, and particularly for this affection, with a reaction to a group of protein that are included in a portion that we call sorry, in wheat, barley, and rye, and are iodine, hordein, and secalin. Now, to be exposed for an individual to one of these proteins and to develop a reaction is generally the result of not being able to identify ingredient is present. So in fact, for all situations, the reaction or the incident, so meaning to be exposed to the protein, for a person who is sensitive to these proteins, is a preventable incident. So in fact, we can prevent food allergy and a reaction of a celiac individual to gluten if essentially these ingredients are excluded from the diet. And speak specifically about celiac disease. Celiac disease is a lifelong medical condition. Again, it is resulting from a predisposition there is a genetic predisposition, and this predisposition leads, when the phenotype is expressed, to having a reaction when the individual is exposed to specific fields. These reactions are generally manifesting by a deterioration of the lining of the intestine, leading potentially to stomach pains or, some kind of abdominal pain, all the way to various conditions that may be associated with that, because when the intestine is deteriorated, you have for malnutrition, leading to conditions such as osteoporosis, lymphoma, type 1 diabetes, increased risk of reproductive problems, but all the way to issues of development, such as delayed puberty in children. There is a proportion of individuals that are known to be suffering from this condition. For celiac disease, the world proportion is known to represent approximately 1% of the population. For food allergy, the statistics in various OECD countries vary between 4 and 6% of the population suffering from food allergies, but the proportion tends to increase, double, up to 8% for children. In the case of food allergies, some food allergies can be outgrown by children. For example, allergies to egg, allergies to milk potentially could be outgrown. For celiac disease, unfortunately, these are lifelong conditions. For both conditions, both food allergies and celiac disease, there is no cure. These situations, there are some research activities underway, particularly for food allergies, where we are looking at potentially desensitization of the individuals through immunotherapies still at the early stages. For celiac disease, to my knowledge, and even though some research is underway, there is no cure, unfortunately. It's a lifelong condition. What is the solution? It's basically to prevent the incidents happening. And to prevent the incidents, the only risk management approach is avoidance. The individuals that are sensitive to these ingredients have to avoid the introduction of these ingredients into their diet. So, to apply avoidance for a person that needs to take away a particular ingredient from, what do they need to have? Information. Information, critical element in order to enable this risk management strategy. And that's why, in fact, labeling has been identified as the approach to support risk management. To work, consumers that have these conditions or that have somebody in their family suffering from this condition, they have to be provided label accurate. It has to be complete. We shouldn't have any hidden sources of allergens, for example, in the composition of the food. The labeling has to be clear and understandable. So a consumer, a mother doing shopping for her family, should not be a scientist to be able to identify that in the composition of that food, there is a protein that needs to be avoided. And it needs to be reliable. We should not have any doubt on the information that is provided through the label. Now, labeling can be regulated, and most food regulatory jurisdictions are, in fact, using the regulatory lever to manage labeling. Now, of course, most countries, as we know, are members of the Codex Alimentarius Commission. And Codex offers guidance on how labeling can be applied. In fact, 2 committees have been working on providing such guidance. The first committee is the Codex Committee on Food Labeling, CCFL, hosted in fact by Canada, and has been working on this, in fact, produced a standard related to labeling of food allergens in 1999. And actually, we will see that this file has been reopened. Another committee has been working on this mostly to support industry, meaning producers, on applying the best risk management strategies to make sure that allergens are excluded from the potential inadvertent introduction into the food, and making sure that industry knows about best practices to manage allergens as a potential food hazard. Now, for gluten, there is another committee that is not listed here because gluten, and to support celiac individuals avoid gluten, celiac individuals are targeting products that we call gluten-free products. And gluten-free products are food for special dietary uses. And as a result, these type of products are managed under the Codex Committee for Nutrition and Food for Special Dietary Uses, CCNFSDU. Now, when we look at what has been accomplished to date, and we will review at least some of the examples, mostly Codex has come up with recommendations on the way to label one of these allergen sources or gluten sources when they are present in food. And the principle is very simple. If you are a manufacturer and you use one of these ingredients in food, you have the obligation to declare that ingredient in food. And if you have exemptions of food labeling, these exemptions are lifted when it comes to these priority allergens or gluten sources. What do we call by exemptions? So for example, when you have spice mixtures, most regulations for labeling in food exempt the declaration of every spice that is included in the mixture. And there is an allowance to use the word spice mixtures without saying what it is. But what if you have some breadcrumbs in the spice mixture, or you have mustard in the spice mixture, or you have sometimes milk powder in the spice mixture? That's a hidden source of allergen or gluten. So that exemption should be lifted, and you don't have to declare everything, but you have to declare at least breadcrumbs or wheat, or you have to declare milk, or you have to declare, whatever it is that you are using, mustard, if it is mustard that is included. So essentially, what the principle is really to declare any ingredient that has that potential and that may be deliberately present in the composition of the food. There's another part that unfortunately hasn't been addressed by Codex guidance until now. And that's the fact that the allergen source may be present not as the result of the deliberate introduction of the recipe, but as a result of an inadvertent presence or as a result of cross-contamination. Example of inadvertent presence. When you produce cereals, and when you produce, I'm going to use an example of a cereal or a source of protein that is generally targeted by celiac individuals. Buckwheat. Buckwheat, al-hanta sauda, I guess. I learned that word, Yani, 2 days ago, so I'm using it. You see? So buckwheat is one of those cereals that is being targeted by celiac individuals. Well, guess what? The way buckwheat is produced, transported, stored, is practically using the same type of value chain as wheat production. So you will be finding in buckwheat elements of inadvertent presence of the other cereals. And this is even allowed by agricultural standards. Another example, oats. Pure oats are actually safe for the majority of celiac individuals. Try to find a source of oat that doesn't contain wheat, barley, or rye. You will find that only if it has been produced under those conditions, because taking, for example, the US agricultural standards, you have an allowance for up to 1% of the inadvertent presence of the other cereals in oats. And 1% is quite a bit of protein in there. So the issue of inadvertent presence, that's an example where it's not regulated right now, or at least it's not regulated by guidance from Codex. The second example is cross-contamination. Cross-contamination comes from where? Well, this is resulting from the potential presence that is not intended as a result of the practices of manufacture. Taking the example of chocolate production, you have a batch of chocolate that contains milk. Milk chocolate is produced, and then all of a sudden, the manufacturer wants to produce some dark chocolate without milk. So milk is not an ingredient. So even though you apply sanitation practices, and we know that generally these sanitation practices, you cannot do a lot of wet cleans in these facilities. So they try actually to push or to dry clean as much as possible. Well, if you try to have the second batch of dark chocolate after the milk chocolate was produced, you will always find an amount of milk protein that is residual, unless you have a dedicated line. And the dedicated line is expensive to have. So in this case, you have a cross-contamination. And in this situation, what has been applied, or at least what has been advised, is to use what we call a may contain statement, may contain on the label. Now, may contain is a good risk management option when it is used judiciously. What does this mean? Meaning that the manufacturer has applied every possible solution to manage the potential for cross-contamination, and despite all that, they cannot exclude the potential introduction of those other unintended proteins. In this case, we put a may contain statement. The problem is we have seen a pervasiveness of the use of the may contain statement. Try to find a food right now that doesn't have a may contain statement. Sometimes, and this is where there is a problem, you will find, and unfortunately, I don't think I have the example here, but I showed it in the training that we had with the colleagues at the military laboratories. You have a food that has 2 ingredients. For example, chicken and salt. It's barbecue chicken. So basically, you have chicken and salt. So how difficult is that? Then you have may contain seafood, fish, nuts, cashew, like the whole thing. In this particular case, don't tell me that you are a manufacturer and you could not avoid the introduction of all these. So when you have a may contain statement that lists the list, and that's what you see on this curve, the more you use the may contain statement, the frequency of use, that results in what? It results in the fact that people are starting to think that this may contain statement doesn't mean anything because it is being used simply, they will say, they are using it to protect themselves from a legal issue, okay? Which means that what they will do is that they will be taking risks. And particularly adolescents will be trying, oh, you know what, it has a may contain statement, let me try that. Oh, you know what, I have nothing, so it's okay. I'm gonna actually try to use it, eat it until there is an incident. So that's what this curve represents. The more you use the may contain statement, the more the probability of incident will increase. And this is potentially an issue. Well, this is not potentially, this is an issue because basically this is a risk management strategy that is no longer useful. So the risk management approach that needs to be followed by industry is really to continue to apply quality management system for every hazard that may be present in the food, but to consider allergens as an important hazard that needs to be managed, and that needs to be managed at the 3 steps of a food safety management systems: the prerequisite programs, the preventive controls, and also making sure that you trace the products, both traceability N-1 at least, and N+1 in case you have a recall, to be able to manage the situation. Generally, what is required is that every manufacturer should develop and maintain what we call an allergen control program that is an integral part of the food safety and quality management system. So the allergen control program is not independent of your food safety management system. It's an integral part of it. The idea here is that you are taking into account the specificities of allergens as a hazard. Part of the elements that needs to be looked at is employee education. So making sure that as part of your prerequisite programs about employee education, people are sensitized to the fact that allergens are a hazard. If you have somebody that brings into the facility a peanut butter sandwich and you're trying to produce, I don't know, confectionery products or, candies or whatever, and people are consuming potential allergens and not applying any sanitation or hygienic practices, you may have a potential of cross-contamination resulting from inadvertent or inattention of the employees. We need also to make sure that, paying attention even to the ingredients, the accurate labeling, that making sure that the identification of the allergen is well taken into account. There should be also, like in any quality management system, a commitment from the upper management of the organization that allergen management is an important issue that needs to be accounted for. So I mentioned the prerequisite programs. In the context of the preventive controls, allergens should be considered as a hazard, just like any other hazards, meaning physical, microbiological, and chemical hazards. Same attention should be made to record keeping, particularly to be able to either trace back or trace forward any potential issues related to the production. This is particularly useful in the context of a recall. And in general, we need to apply a risk analysis-driven approach in the context of allergen management. Now, when you are applying a risk-based approach, one of the elements that you need to rely upon is the identification of the hazards. And how do you identify the hazard? It's through analysis. The analytical tools help you to identify or to confirm the presence of the hazard. Now, an example of a risk-based allergen management program that is developed by food business operators. I'm not going to go through it. I mean, you can have the slides. By the way, this approach, in fact, is inspired by the guidance that was developed by the Codex Alimentarius Commission and mainly by the Code of Practice to Manage Allergens in Food that was adopted in 2020. Now, going back to Codex particularly, and the international guidance. So, as I mentioned, the Codex Alimentarius Commission led the way in developing guidance on how allergens should be managed, mostly focusing on labeling. And the main standard that was developed was the 1999 standard, calling for the identification of those priority allergens and a mandatory declaration on the label of prepackaged foods for any allergen that may be included in the recipe. Now, for gluten, the standards are actually dating of the late 1970s. So, in fact, since the 1980s, we had a gluten-free standard adopted by CCNFSDU. The only difference is that the gluten standard has been amended many times. And this was actually a good thing because it took into account the development of the scientific information related to celiac disease. One of the elements of the scientific information was the fact that gluten reactions are threshold-based. And the issue was to be able to identify what the threshold was. In 2015, the latest development of the standard the gluten standard relied upon the scientific information that tells us that celiac individuals in majority would be able to tolerate up to 10 milligrams per day of gluten protein. So that's the tolerance that was identified globally. This is based on a pivotal study that was done by Katasi and his group. So in fact, in this particular case, the gluten-free standard was amended to identify a target level for food that can put on their label gluten-free statement. And this threshold now, this now has become an analytical threshold, is 20 ppm. It was identified that 20 parts per million or 20 milligram per kilogram of gluten protein would be, if it is present, so this is the maximum level, if they are present in gluten-free foods, would achieve the protection of the majority of celiac individuals. That's essentially what was identified. Now, the gluten standard is the only one, if I compare it with allergens, even though, I mean, I should not be comparing, but let's say it's really those ingredients that induce a reaction. The gluten standard in Codex is the only one where there was a dedicated guidance for an analytical method. And Codex, under now the advice of CCMAS, so the Codex Committee of Methods of Analysis and Sampling, which we will be discussing later during the day, has actually a guidance on the methods or the performance criteria for some methods that could be used to detect gluten markers. Basically, Codex has identified a type 1 method. Now, type 1 is very important. What does this mean? Type 1, meaning that you have one target. It's the only type of method that should be applied to determine the markers related to your standard. In this case, the marker related to your standard are gluten protein. So this is an exclusive type of method that is relying on enzyme-linked immunoassay with a particularity, the R5, it's called the R5 antibody. Now, this R5 antibody, it was developed by Mendez. That's why we call it actually the Mendez antibody. And it targets a pentapeptide that is repeated at a very high frequency in proteins that are known to compose the gluten source in both wheat, well, not both, in wheat, barley, and rye. So in gliadins, secalins, and hordeins, you will find essentially this pentapeptide. And basically, many methods and many proprietary test kits have been developed as a result of this guidance developed by Codex. As I mentioned now, Codex reopened the file of food allergies, and in 2018, actually, a set of work has been put in place, mostly resulting in the guidance to food industry in the form of a code of practice developed by CCFL. This is the code, CCFH, the Codex Committee on Food Hygiene. Code of practice on food allergen management for food business operators that was adopted in 2020. Basically, it's again, calling for a harmonized proactive approach in managing allergens, calling for food business operators to develop policies and procedures to identify allergens and also to put in place control measures, including risk communication measures, which is essentially the label, improved labeling. These are the components of the areas of intervention that were called upon by the Codex, the updated Codex text. And you see various components of food safety and quality management, including training of operators. Now, most recently as well, Codex has requested scientific advice from its expert bodies. So generally it's JECFA that is the expert body, the Joint Expert Committee on Food Additives. But in this case, they had to create a special expert consultation on food allergens. And they asked some specific questions to this expert group. One of the first questions was to say, in 1999, we developed a list of priority allergens. Is this list still valid? should we consider the list of priority allergen the same one as we had in 1999? The second question is, we know that food allergy reactions are threshold-based. Now, this is not about gluten. This is about allergens. What are the thresholds for food allergens? Because we didn't have any information at the global level. And the third question is, can you help us manage precautionary labeling? Because we need some scientific foundation for this. It's a problem, this precautionary labeling. How can we fix that with scientific evidence? This is essentially what this expert group worked on. And as I said, we will share this presentation with you. And you will find here, in fact, 4 reports that were produced by this expert group trying to answer those 3 questions. Now, one of the first things that came out is the update to the list of priority allergens. Basically, in order to do that, this expert group reviewed the criteria of what makes an allergen a priority allergen. They identified 3 requirements. The allergen or this ingredient has to be associated with a high prevalence of food allergies. So the prevalence at the global level. Second element, the reaction associated with the consumption of this ingredient needs to be a severe reaction. So meaning that if the only thing that I may have is itching or urticaria, yeah, it's important, but you know what? Maybe not a priority allergen, okay? So in this case, anaphylaxis, etc., would be one of the, conditions that we would be looking at. And the third criterion is what we call potency. Potency meaning what? Meaning that a small amount of the ingredient may induce a high reaction. So in this particular case, it is important. It can be a priority. So as a result of this, the list was updated and soy, which was part of the priority allergens, was taken out. But sesame, which is essentially we're going to be hearing about in a later presentation by Dr. Touma, sesame was introduced as a global priority allergen. This expert consultation also identified some proteins that needed to be put what they call the watch list. And this is because they wanted to acknowledge that the conditions of production of food has been evolving. And as a result, there are new proteins that are being introduced in the composition of a number of foods, such as pulses, al-buquliyat, that are introduced in this composition. Insects are now being introduced as part of the production of food, but also other fruit, such as kiwis, identified also mostly in relation to potency in this particular case and even potential prevalence of the allergy. The second question they tried to address, and this is essentially the proposed threshold list that they came up with. The threshold list is actually the result of what they called a clinical studies that they have analyzed. They've tried to come up with what they call a reference dose. And this reference dose is, in fact, the dose that would protect 95% of the individuals. Meaning, at this dose or less, at this dose or higher than this dose, basically only 5% of this dose, at this dose, sorry, I'm trying to think now. It's at this dose or less, only 5% of the individuals would react. 95% would be protected. Okay, and that's what they call actually the EG5 dose. Okay, so you protect 95%. And if you have this dose, basically you would have only 5% of the population that would react to this particular dose. They also came up with an approach to help manage what they call precautionary labeling. And this approach relies, in fact, on a risk assessment. What they're asking is that a company should not be using a may contain statement unless it is resulting from a risk assessment. They assess the risk associated to their manufacturing conditions, and they see if the risk warrants that they can put a may contain statement, including, in order to do this risk assessment, that I need to analyze either the environment to see if there is residues of allergens, or products to see again if there are residues or sources of cross-contamination for the allergens. And on the basis of that, I can try potentially to make a risk assessment and determine whether or not I should use a may contain. Generally, the approach is very simple. You have the reference dose. If your risk tells you that the intake is higher than the reference dose, you should put a may contain. If your risk assessment tells you that the intake of the protein is lower than the reference dose, you should not put the may contain statement. It's a very simple approach. But what this tells you is what? You need to determine in a quantitative fashion, in some situations, the presence of the allergens in order to assess the risk. So that's actually what now Codex will have to be looking at. And the next session of CCFL, which will be taking place, in fact, in late October in Canada, will be looking at this to try, first of all, to endorse these lists, the updated lists, but also to come up with an approach, precautionary allergen labeling. Now, of course, here, I'm trying to put the importance of analytical methods. Analytical methods will need to be available to address priorities of risk management in the context of allergens. Now, thankfully, since the 1990s, we have so many analytical methods. I mean, if you compare, and I guess I'm old enough that I can attest to that, you know, in 1996, there was not a single test kit for the analysis of allergens. There wasn't a simple, even a single instrumental technique that was dedicated to analyzing markers of allergens. Today, you have even AOAC approved methods, both for instrumental techniques as well as test kits. The problem, though, is, can you tell me, for example, what is the JFDA? I'm sorry, actually, I'm putting JFDA here on the spot. What is the JFDA approved method to test for undeclared peanut? Or what is the SFDA, you know, approved method to test for undeclared peanut? I don't have that. And then how can you expect that a manufacturer would be choosing or targeting a particular analytical method if the regulator is not providing that advice? So we need to standardize essentially these approaches, and to standardize these approaches based on agreed upon performance criteria for the methods, and using these performance criteria to analyze the performance of available methodologies and try to see if we can put them as potentially standard or reference methodologies that could be used by regulators and potentially by industry. Here, thankfully, AOAC has done a lot of work in this regard. And in fact, we have now performance criteria requirements for the acceptance of methods developed both for allergens and for gluten through AOAC. The question is, can we use this particular guidance and, in fact, develop an Arab compendium of allergen testing methods as part of the Arab compendium of methods? And this brings us to something that we would like to do in conjunction with the Arab section of AOAC International, to leverage the AOAC processes, to look at available methods that are being used by the community of practice of allergen testing, and to then try to develop a process for acceptance of methods that fulfill certain requirements, and actually to make available the way these methods perform in the form of scientific articles that are accessible to all. And actually here, it's my pleasure to announce that we will be using this journal that will be created and implemented by the Arab section of AOAC International, the Journal of Official Food Analytical Methods for the Arab region, as the conduit for that. By the way, I mean, the journal is, the link is created. So if you put jofamar.com, you will be able to get that site. But right now there are no articles because we're just starting this. So we're hoping that this will be a conduit for that. So let's hope that we will get the collaboration of the analytical community from the food allergen team here, and that we will get this work in motion. So I hope this presentation set the stage, and I'm seeing that I probably exceeded my time as a chair of the session. I'm not giving the great example here for this presentation, but maybe I'll ask my co-presenters here that they will help me catch up back, or we'll see how we can manage the panel discussion. Thank you very much again for your attention. So we'll hold questions for now, and we'll move to the subsequent presentations, and we'll have a Q&A session subsequently. So it's my great pleasure to invite Professor Hani Al-Dmour, Professor of Food Science and Technology at the Department of Nutrition and Food Processing at the Al-Balqa Applied University in Jordan, and Professor Al-Dmour will be sharing with us his perspectives on gluten testing in food. Thank you.
First, I said, Alhamdulillah. He didn't talk about my presentation. He talked about everything.
That's great then. It's well coordinated.
Yes, yes. Alhamdulillah. Assalamu alaikum. أنا بسعدني أحكي two reviews.
Peace be upon you. I'm quite happy to talk about two reviews about the gluten testing and also the creative officers from the army. The first review is our colleagues from the Jordanian Armed Forces who had adopted the idea in 2019 during the Corona or the COVID pandemic. We encountered a problem with the PT. I consulted my colleagues such as Rana Rawashde, and we had a lot of work with the anti-drug smuggling department and the customs and the border forces, etc. So I talked to the Jordanian Armed Forces. I attended a meeting with Mr. Dana Marashd and talked to them about adopting the idea of making the PT. So they adopted the idea, they were convinced, and they decided to exert all the efforts in order to provide or to make the PT available, because the PT is quite expensive. We lose our money and hardly can provide enough PT. So without Lana Marashd, Lana Rawashde can confirm my word, and I'm quite proud of the level of collaboration between us and the Jordanian Armed Forces. They were quite enthusiastic about the idea during the discussions, and this was the first review. However, the second review, thank God, my colleague here did not talk about it. He's a genius. He's quite a genius. However, my presentation is going to be on testing. I reviewed the journals and I reviewed all the papers published. Dr. Samuel talked about the gluten testing. I think. Anyway, Dr. Samuel talked about proteins. However, regarding gluten, we are interested in the gliadin group and the glutenin group. These two cause the problem. However, you can find gluten in cereals and in barley. And the problem lays in the pollution. You won't find oat without it. So according to the specifications, it's allowed to have a certain percentage of gluten. So second, Dr. Samuel had talked about this topic as well. We have a gluten intolerance and we have celiac disease. Both of them, they need gluten-free diet. There is no other solution. You need to provide gluten-free nutritious products. The difference between them is quite minimal. However, at the end of the day, in order to help the patient who has these cases and in order to stay alive, they need to have gluten-free food products. However, if the ratio is higher, or if there is, as for the celiac disease, if the food has more than 20 milligrams, then it's not allowed. And what I would like to say now is a review. The testing review for gluten, it takes the form of researches that are published. These researchers had published the most important methods around the world, and most of them are currently used. And as for the gluten testing methods, can be summarized or classified into 3 methods. The first one is genome, which is the PCR, mainly, and the methods that rely on the proteomic, which rely on studying the protein and gluten. And the third method is the immunoclinical, which rely on the antibodies and antigens, and most importantly, the ELISA. The PCR, the first method. Yes, this is slide. The PCR in 1993 was the first research published by the use of base assay, meaning the PCR method. After that, the researchers found found interference or false results. So researchers, since they have a scientific mentality, one of the researchers in 2001 had developed the method and inserted the quantitative method using the PCR. By the way, I'm quite aware of most of the laboratories in Jordan. These methods are already in use. If you want me to share this paper with you, I can just let me know. I have the reference and I have the paper with me, and maybe you can review it in order to start using these methods and try to have them used in the laboratory. So in Jordan, we use the ELISA mostly. I don't know I don't know if some laboratories had adopted the PCR method. Another researcher in 2003 had developed the method using the PCR and inserted the immunin PCR assay in order to detect the gliadin. After that, in 2011, on that year, another research that was published by developed the method and inserted the IV sensitive PCR. This advancement helps us in using certain different applications. So these are the most important developments on the PCR for testing gluten. This paper is published and accredited. As for the PCR methods, I tried to look for the advantages and disadvantages. And when I reviewed this paper, I found that some researchers had worked on ELISA PCR. Other researchers had also used the HPLC and the mass spectrum and made comparison with the ELISA. So those studies were quite fruitful, and you have to realize these are only researches, but had the results adopted in the real life. I believe that if you are interested in executing the methods stated in the paper to be used as a reference, I will be happy to share it with you. In the paper, they say that it is PCR is more sensitive, and it did not present false results. When you draw a comparison with the ELISA, it gave 2% of false results in the detection. So we can say that the PCR currently is being applied, and the PCR could be a method to encourage the PCR could become prevailing, and I encourage people to use the PCR in order to detect gluten contamination. And remember when I said about the methods that rely on protein? So those methods, yesterday we talked about them a lot, and when we talked about the matrix, the mass spectrum, there are papers published using the mass spectrum for qualitative and quantitative testing of gluten, and it provided excellent results. However, are these methods approved from A to Z? I think, yeah, could be yes, could be no. However, this research is done by respectable researchers, and probably in the future these methods could be approved by the AOAC. So the paper is published, and we trust that when the paper is published, our confidence increases, and the results stated in the paper are can be compared. The LC tandem mass spectra MSMS, it was also used for gluten, and these are the devices used in order to estimate the gluten, and therefore this means that we have new horizons. Sometimes by changing certain conditions and making slight changes, we can use the devices used to analyze and to analyze the pesticide residues to be used for gluten testing.
كونه بروتين تعرفوا اكيد يعني كان في مش يعني بجوز كان في development
So there were some development in the separation process between the proteins. So you have seen in slide number 2 that there were a number of proteins. So when some proteins unite with other proteins, they make certain, they behave in a certain manner. And here, in this study, they use the gel in order to separate the proteins. This means that this step had increased the accuracy and sensitivity and the results of gluten testing. This method has advantages and some disadvantages. Here, this bullet is highlighted in yellow. It talks about the disadvantages where non-protein, there might be interference between non-proteinic gluteins. This means interference. In this method that I like, because the infrared it can be used, which is cheaper than the LC. I'm talking here about 40,000 or 30,000. And the infrared technology, if the information, if the method is as easy as I reviewed in the paper, the only thing you need for the infrared is to do calibration, and it is a nice way to be used. I believe this device is available in most of the laboratories, and the method of using the infrared is indicated in several published papers. The last method is the it's in the immune disease, immune diseases. So according to the paper, they used two mechanisms in the ELISA, which is the sandwich ELISA. This is the technique. And the other technique is competitive ELISA. It's quite known. Now there are new immune methods, which is called the rapid. It's mostly used in all of the laboratories. In my laboratory, we use the ELISA and we use the rapid. And the rapid is AOAC approved. In the picture, you can see the expert or the scientist analyzing using the ELISA, and we can use these methods, the antigen antibodies, and we use the sandwich ELISA. Of course, each method has its own pros and cons. As Dr. Samuel had said, the codex had recognized, and actually it means that they have some kind of respect to it, because the codex is our constitution. In the field of food, codex is our constitution. And accordingly, you can rely on this method as an acknowledged method for gluten testing. And since all of us, or most of us, have the ELISA, so I hope that the Jordan Army can make PT of gluten via the ELISA. We can do the PT on gluten by the use of ELISA. As it's not rapid test is an addition to have extra readings. Here, the heat processed foods create certain changes, and they might cause some problems for the gluten testing, and we can avoid these issues to have more accurate results. On this slide, you would find the challenges in gluten detection. In the gluten testing, first, we might have food contamination, especially in the laboratory. This is why we need accreditation for gluten testing, and we need to have accurate results. When we gain the accreditation, our work will be more accurate with high sensitivity and accuracy, and we will be able to avoid the problems of interference. Anyway, I would like to conclude this presentation by saying that there are new methods that are promising. The infrared is easy to use or to handle, and the ELISA as well. Dr. Samuel said that in the codex there is a reference. We can adopt it in our laboratories and make PT for the gluten testing. Thank you very much for your good listening, and I hope it was an easy to grasp presentation.
Thank you so much for this great overview on gluten methods. And again, as mentioned, we will have the opportunity to take questions subsequent to all the presentations. Now we're going to be moving to back to allergens and taking an example of an allergen that is known to be quite prevalent in the Near East, and that's sesame. And this is an allergen that has also been introduced more recently into the list of priority allergens by the Codex Alimentarius Commission. I would like to invite Dr. Joseph Touma, researcher at the Lebanese Agricultural Research Institute, the LARI, and also holder of a PhD from Laval University. He's going to be sharing with you some of his work on the occurrence and risk assessment of sesame allergens in Oriental type products on the Lebanese market. Dr. Touma, would you like to use this or this one?
Okay. السلام عليكم. استكمالا لمحاضرة الدكتور بروفيسور سامويل سوف أقوم بعرض a case study about occurrence and risk assessment of sesame allergen in Oriental type product on the Lebanese market. سوف نقوم. Okay. رح نقوم بعرض an overview.
So we will be having an overview of the sesame, its prevalence in Lebanon and the Arab region. So allow me to begin. So sesame is a global agricultural crops.
2.3 million to ton in 1994 to more than 5.5 million in 2007. Several countries follow the allergen labeling requirement of the Codex General Standard for the labeling of prepackaged food in order to protect the health of consumer allergy. And this allergen, when they are used as raw material, they are known as priority allergen, and we are focusing about sesame. Report of sesame allergy appears to be on the rise due to the increasing popularity of vegetarian dishes and cooked meal, and the use of sesame in fast food and international baked products. The immunoglobulin Ig-induced sesame allergy began early in life and may persist for life in 80% of patients. The high consumption of sesame may increase the risk of allergy due to the accidental ingestion of sesame seed and consumption of prepacked food that should not contain them. And here we are talking about cross-contact, and Professor Sam talked too much about this. Sesame allergen can trigger serious reaction in people with allergy, which can lead to a life-threatening anaphylactic reaction. Globally, the prevalence of sesame allergen is estimated between 0.1 and 0.2%. In Kuwait, the prevalence of self-reported sesame allergy among young adults has been determined as 0.46%. In Iran, the estimated proportion of sesame-induced anaphylaxis is high among all cases of anaphylaxis. Sesame is considered the third allergen responsible for food-induced anaphylaxis in Saudi Arabia. In Lebanon, sesame is a major consumer. Lebanon is a major consumer of sesame seed and its byproduct, and is ranked third after milk and egg in Lebanon, and young children are particularly affected. In 2015, following a survey conducted by telephone, the self-reported prevalence of sesame seed allergy in Lebanon was estimated at 5%. Situation in Lebanon. We know that we have 40% of the Lebanese agri-food established are involved in the production of baked and confectionery product. And Lebanon is a major exporter of sesame-containing product, especially to Australia, to Canada, to USA, and to the Gulf countries. Probabilistic risk assessment of food allergen on packaged or unpackaged food product had not been carried out in Lebanon and even in the Middle East and even globally. So our objective was to conduct a survey on the occurrence of sesame in the Lebanese food products, and then to have a survey on food consumption in Beirut school, private and public, and to determine the exposure and then to determine the risk. The methodology of the risk analysis adopted, we are based here on risk analysis, تحليل المخاطر, which has the risk assessment, risk management, and risk communication. And we are focusing here on risk assessment to estimate the risk from the consumption of product containing sesame protein. Here is the concept of the probabilistic food allergy risk assessment. So first, to determine the risk, we need occurrence data, and then we need consumption data. The consumption data with the occurrence data, we will determine the allergen intake distribution. And we need scientific literature about individual allergic. And these, with the distribution allergen, will determine the risk of allergic reaction distribution. So the study was conducted in Beirut on bakeries and from food industry. In total, we have 1,270 samples were analyzed. We have three types of products: unlabeled product, like bites, المعجنات, petit four, croissant, and صفوف. And we have labeled product with and without precautionary allergen labeling, اللي حكي عنها الدكتور سامويل, precautionary allergen label. And this product was bread, cake, coconut, French bread, toast, cake with milk, and the product from industry, it was jam and spices. The method used, it was the ELISA sandwich, which was validated in our laboratories. And for the sample preparation, we have ensured good homogeneity of each sample to be analyzed. The kit was provided by RBioPharm, and the limit of detection of the kit was established as 0.14 milligram sesame. The dose obtained were converted from milligram sesame to milligram sesame protein by multiplying the amount of sesame by 0.17. Sample with a concentration of sesame protein above LOQ was considered positive. The absorbance of the analyzed sample is determined at 450 nanometer in a spectrophotometer reader, and the resulting concentration is compared to a standard curve. For the result, here we are talking about a product, unlabeled product. SFOF was the higher, all the sample of SFOF analyzed was contaminated. 40 من 40 عينة SFOF. So 47% of the product in total was contaminated. For product labeled with PAL, here we have the CAC was highly contaminated, and 43 of the sample were containing sesame protein. For the product labeled without PAL, here also the product CAC was highly contaminated, and 26% of the sample was contaminated, all and all. Concerning the consumption data, we made a semi-quantitative survey of the frequency of food consumption in the private and public school in Lebanon, targeting two categories of age. The first one for between 4 and 11 years, and the second one between 12 and 18 years. It was conducted in Beirut, where 40% of the population are located. In total, 1,156 questionnaire was done. So till now, we have the occurrence data with the consumption data, and these will lead us to the allergen intake distribution. Now we need the scientific literature, and these data are taken from different articles around the world. So we had data for about 40 allergic individuals, and these data allow us to construct the dose response curve for sesame. So as we mentioned, we have the allergen intake distribution, and with this data, we'll determine the risk of allergic reaction due to the consumption of product containing sesame protein. The method used, so the probabilistic risk assessment, it allows better use of the existing information. The software used was ARI statistical software, and we used the two-dimensional Monte Carlo simulation method. And the risk assessment assumes that all individuals in the simulation are allergic to sesame. The result for the consumption of product, unlabeled product, here the SFOF was the higher risk. That means that on average, 203 out of 1,000 eating occasion for the children, and 231 out of 1,000 eating occasion would lead to a reaction in these two categories of children and adolescents. For the product labeled with and without PAL, here we have the product CAG with a higher risk. As recommendation, as conclusion, so we need to do a better risk management in food industry and bakeries, limiting economy class. The food industry and regulators should adopt a transparent and risk-based approach, and need for regulatory and/or voluntary risk management measures, targeting unlabeled products especially, and strengthen risk management measures, including regulatory measures related to allergen labeling in Lebanon, not only for use of PAL, but overall to enhance compliance with the requirement of declaration of ingredients on food labels. And thank you for your attention.
Thank you very much, Dr. Touma, for this presentation and for catching us up, I think. We're going to be now going back to gluten analysis, and we have the privilege to have with us Dr. Stefan Schmidt. I think he's online. Stefan, I don't know if you can hear us.
Thank you for giving me the opportunity here to speak in front of the audience, although I'm not in person there, but modern technology is enabling us to do it in that way. I would like to give you a short overview about the official method for gluten analysis and the history from the beginning to today. So we will speak about Codex Alimentarius reference method. We will speak about the official method of analysis for gluten at the AOAC, which is in fact more or less the same. And last but not least, I will talk a little bit about the importance of so-called incurred samples for the validation of methods. Let's start a little bit with the history. Here on the right side, of course, it will be probably a little bit small to read everything. You see the history starting in 1983 with the first Codex standard for gluten-free food. At that time, we had really high limit values, which are much lower nowadays with new assays. The first ELISA method was the so-called SCARIT assay or SCARIT ELISA, which had an LOQ of 150 ppm, 150 milligram per kilogram. And this assay was not able to quantify gluten from barley. But then in 1999, the working group of Mendes in Spain introduced the first sandwich ELISA based on R5 antibody, which was an improvement because this assay could detect gluten from wheat, rye, and barley and showed a very low or very good sensitivity with a low detection limit. This assay was then improved in 2002 with a specific cocktail solution to extract gluten from heat processed samples. In 1999, it was usual to do a easy or simple ethanol extraction, but it was clear very soon that this ethanol extraction was not sufficient enough to extract gluten from heat processed samples. And this was then published in 2003 in the European Journal of Gastroenterology and Hepatology by Mendes and his working group. He introduced here the sandwich ELISA based on the monoclonal antibody R5. And he focused in this work especially on analyzing cooked foods, heat processed foods, where the gluten is embedded in the matrix in a way that it cannot be solubilized by ethanol only. So he introduced here a special cocktail extraction procedure in combination with the R5 ELISA to resolve the gluten proteins from the matrix. And this method was then also in 2008 brought to the Codex standard, the Codex Standards 234, which is the collection of official Codex reference methods. Here you see this publication from Mendes, which is announced as the Codex reference method type 1, which means this is the method which defines the amount of gluten. We do not have any other reference methods for gluten. You have heard in the lecture this morning that there are, of course, also other methods to detect and analyze gluten, but there is no other official reference method than this R5 ELISA. So this R5 Mendes method in combination with this cocktail defines what is in a sample regarding gluten. So the combination of R5 ELISA plus cocktail is the so-called reference method, the Codex Alimentarius type 1 method, as it is mentioned in the Codex standards 234. In addition to this Codex standard, we have the standard 118, which is the standard for foods for special dietary use for persons intolerant to gluten. These two standards in combination tell what to do regarding gluten analysis. In the standard 118, you find the limit to differentiate gluten-free food from gluten-containing food. The limit value is 20 milligram per kilogram per definition. We see here also a definition of prolamins, which can be extracted by 40 to 70% of ethanol. From heat processed food, we have learned ethanol alone is not sufficient. So there we have the combination of this Mendes cocktail with 60% ethanol for the extraction of the prolamins. The prolamins is a specific part of gluten. If we look at wheat prolamins, then they are called gliadins. If we look into rye, it is the secalin fraction, and in barley, it is called the hodein fraction. You see also here oats mentioned. The prolamins from oats are avenine. However, these avenines are immunologically different compared to gliadin, secalin, hodeins. So therefore, most celiac patients do not show any adverse reaction against avenines from oats. And you see here the note, oats can be tolerated by most, but not all people who are intolerant to gluten. Therefore, the allowance of oats that are not contaminated with wheat, rye, or barley in foods covered by this standard may be determined at the national level. If you look nowadays around the world, most countries allow oats for gluten-free diets. There are only few countries which look especially for oats. This is, for example, Australia, but in most other countries, oats is allowed as a part of gluten-free diets. Since the prolamins are only a part of gluten, there must be used a factor. Prolamins, by definition here in this Codex standards, is 50% of gluten. Therefore, if you measure with a gliadin assay the gliadin content of a sample, you have to use a factor 2 to calculate then for gluten. And in the standard, you find also mentioned the R5 Mendes method as the method to determine gluten. If we go further in the history of R5 methods, then you will see that we as a biopharm have introduced an R5 ELISA to analyze gliadin already in 2002. In 2008, we got the official method status from the AOAC. So this method was validated and approved by the AOAC. And in 2012, it became then the final status of official method as of analysis by the AOAC for foods. This was at that time usual to call an ELISA method as an analytical tool for something in foods. However, a little bit later in 2016, the AOAC changed its rules and it decided, yeah, we have to be a little bit careful. We cannot say foods in general. The studies were made with rice and corn-based matrices. And therefore, in 2016, the official method from the AOAC was changed to rice and corn-based matrices. So people were confused at that time. Can we use the R5 ELISA only for rice and corn-based matrices? Is it not allowed to use it for food? Of course, it is allowed, but you have to validate it. Due to this confusing situation, we decided to do another international collaborative study. It was then already a third study that we did with our ELISA in 2020 with a wide range of different matrices to demonstrate that the method is applicable also to foods in general. And at that time, the AOAC changed its rules also regarding the samples that need to be tested. We have learned already that heat-processed samples are difficult and behave different compared to native samples. And therefore, the AOAC has included in between in its validation guidelines that incurred samples need to be tested. Why is it so important? For this, we have to remember our lessons of biochemistry that we have learned in the past. Proteins show different structures, primary structures, secondary structures, and so on, as they are shown here. And it is very important that an antibody, which is used in an ELISA, like the R5 antibody, only detects one specific epitope. So it may react with a primary structure, it may react with a secondary structure, but not with both. The R5 assay antibody detects primary structure from gluten. What is going on now when you are processing ingredients, foods, which show in the native behavior certain structure? Here you see just a model of a native protein. If you process this protein by heating, for example, it will change its structure. It may lose different structures. And if you go further with processing, highly processed samples like hydrolyzed samples have only peptides from the original gluten included then. So if you look at the antibodies, you may have antibodies, as I said, which react with primary structures. These structures will be present in all different types that are shown here. But an antibody which reacts with a secondary structure, like this red one here, will not be able to detect these structures in a hydrolyzed product, but perhaps in a heat process. And just another example, an antibody which reacts with this beta structure, if it is destroyed or changed in a heat processed sample, it will not react with this then in a heat processed food. So you always have to think of what do I test and what is the assay able to detect. Of course, this is a gradually process. The longer you cook, the longer and the higher you heat a food, the more these changes will be present. So for the gluten, we have this situation here. We have ingredients which are perhaps contaminated with wheat, rye, or barley. With food processing, for example, production of pastries, noodles, you may change the proteins, the gluten inside these ingredients. So the food, the ready prepared food shows a different reactivity compared to the ingredients in an assay. And then finally, for the analysis, you have to extract the proteins from the food, which is complicated. Therefore, the special extraction cocktail was developed by Mendes, just to find only the targets that we want to get, the gliadins, saccharins, hordeins, so the gluten proteins. And both the processing and the extraction will influence the structure of these proteins and therefore their activity and the recovery in an assay. Therefore, it is so important to look when you validate an assay also to incurred samples, not only native raw materials, also the combination of different ingredients, processing of foods is important. And so we included in our third study, international study, different food samples, in total 19 different real food samples, not only dry material, also humid, moist material, which is difficult to send through the world, to have it in the same way in all labs which are taking part in this study. The foods were prepared independently at a university in Germany. The foods that this company prepared, you can see here on the next slide, and you see a high variation of different procedures, different time of processing, different temperatures of processing. We also looked, see it in the middle of the right table. We also looked for microwaving because there are there's one publication at least telling that microwaving may have an influence on the recovery. We could not see this influence here in our study, but therefore we included microwaving as well in the study. So all in all, we sent out to 14 laboratories closely 900 samples, which were then tested here in Austria, Canada, Finland, Germany, Ireland, Italy, USA. So worldwide, these samples were sent and tested for the content of gluten to show that the R5 ELISA from a biofarm is able to detect gluten also in processed samples, and that also were laboratories all over the world are able to find comparable results. And that you can see here on this slide, the recovery for all these samples were mostly between 80 and 120%, depending on the food matrix that is analyzed. There was only juice, which showed a little bit lower recovery of 60, 70%. And also chocolate is always a tricky food matrix. So the chocolate cake also showed only 60% recovery in this study here. As reference material, we used two different types. One was the PWG gliadin, which is already available for many years. And we used a new flour, which was prepared by the organization of Monica. But these did not differ from each other in this study, as you can see here with the light and dark gray bars. So finally, because of these good results, the AOAC granted our essay again the claim measure of gluten in food because of this wide range of food which was tested in this study. The AOAC decided, yes, it is not only able to detect gluten in rice and corn-based food, it is also able to detect gluten in food in general. The results of this study are published as open access. You see the link on at the end of this slide. So this was the combination tested in the study, the R5 gliadin ELISA, real screen gliadin ELISA, in combination with the Mendes cocktail. So this combination is not only the official method of the AOAC, but also the Codex Alimentarius reference method, because it's an R5 ELISA, and the cocktail from Mendes is used for the extraction. However, there may be situations where this combination of cocktail and sandwich ELISA is not the best choice. We have seen hydrolyzed food where you have only peptides can be also a problem with analysis. And the small peptides are not able to build up a sandwich in a sandwich ELISA. Therefore, you need a competitive ELISA setup. So we have also a competitive ELISA in our portfolio for this special kind of foods. Additionally, sometimes it is perhaps important to get a fast result for hygiene testing, for example, to see, is your equipment clean? Do you have a contamination in your lab? So for this, we have here a rapid assay, which can be used for swabbing the equipment that you use, for example, for analysis, something that we always recommend to check for cleanliness in your lab with such an assay. This is also a validated method by the AOAC. It's a performance tested method. So to come to the end of my presentation, a summary, keep always in mind what you are analyzing. Is it just a raw material? Is it a processed food? Is it a ready prepared food? This will have an impact on the protein structure depending on when does a contamination occur. Was already the raw material contaminated, then all the food processing steps will influence the recovery of the gluten. If only the final product was contaminated, then of course the situation is a little bit different. Therefore, you have to think of what is tested in my lab, what can be the impact on the protein structure and therefore also on the recovery. And that was the last slide from my presentation, and I thank you for your attention.
Thank you. Thank you very much, Stefan, for this great overview on the two applications for both hydrolyzed and non-hydrolyzed types of food and the measure of gluten. I'm going to invite you, Stefan, to stay with us, if you don't mind, for the questions and the discussion period. And as the questions may be in Arabic and some of the discussions may be in Arabic, you know that simultaneous interpretation is provided, so feel free to switch to the English channel if some of the discussions are in Arabic. إذا نمر إلى فترة نقاشية ولو تكون مقصرة لهذه
Now this session will be dedicated for Q&As. I would like to invite all the participants with us here in the session, Dr. Tuma, Dr. Stefan, Dr. and the representatives of the SIAC Society from the Hashemite Kingdom of Jordan, and also Ms. Hamaida representing the Jordanian Armed Forces from Jordan. We will receive your questions or comments from the participants, whether physically here with us or via Zoom, and kindly speak through the microphone so that the interpretation will be provided. Good morning, everyone. I'm Dr. Rawan Hayari, the director of the Laboratories Directorate from the Food and Drug Association. The speaker has switched to English.
Available advices related to the methods of analysis and related issues regarding to gluten, which take us to a mutual recognition between the health authorities later. يمكن بحب أحكي إنه المواصفة القياسية الأردنية.
I would like to talk that the Jordanian specifications and meteorology association had worked with our partners from the Jordanian Armed Forces, and that the gluten testing should become obligatory or mandatory. So in order to receive a certificate, the Jordan Specifications and Meteorology Association is going to conduct a gluten testing, and accordingly the laws and regulations will be amended to enforce this decision. This is a very good news. Because this will also generate data regarding the conformity of processed food and also food products, and whether they fulfill the requirements, and those requirements, whether we're talking about the requirements and conditions of the Jordan Specifications and Meteorology Association. We should enhance collaboration between the competent authorities and also we can collaborate with the Chamber of Commerce and the Chamber of Industry to be on board. God willing, we hope to establish this kind of partnership and collaboration between the competent entities, and this can be among the activities that we will organize to facilitate this kind of collaboration. If you would allow me, good evening, everybody. At the beginning, I'm quite proud to be a member from the Jordanian Armed Forces. Some people might think that the Jordanian Armed Forces are only specialized in defense activities. However, we are always in the frontier of any initiative to achieve advancement in every discipline and field. The painful case of the patients who are not heard in the military laboratories, we played a humanitarian role. We overcome all the barriers, and we communicated with the competent entities, the Ministry of Agriculture, the Ministry of Health, and the Ministry and the Chamber of Commerce, and the Jordan Association of Specifications and Meteorology, and all the relevant competent entities to contribute. However, the training that took place 2 days in the military laboratories, we have the scientists realized that we still need to learn more. And yesterday, when I was working on my slides, I found that the ISO issuing assess studios is over 20 and I believe we can work on ourselves more, and we ready to fulfill requirements, and I believe that we are who are in service. However, we conduct monitoring, and the Ministry of Health also do monitoring, but our data and regions are not unified. We should have a database. I believe we need to have unified statistics our needs. When we look for a technique or a rapid regulators to be on board with us. It's currently the Jordan Association for Rheology working on regulatory until to control I hope that the regulations will be endorsed very soon in order to prevent any manufacturer or from manipulating the food safety on our national soil. Good evening. Good evening. I'm the head of Celia Patients. The doctor said a very valuable information and care for her for the efforts exerted field. And the professor earlier talked about the Celia Patients Society, which is by us in Jordan. The establishment of our society, we've been monitoring the efforts exerted by the competence, the Jordan the are first only I would like internal about this efforts and adopted our issue and to bring the testing kits. There was a program with the FDA. They used to do this work or collect the samples and you used to send for testing Jordan Association for Standards and Meteorology. However, within the next months, we will overcome all the challenges because on this moment haven't been able to crawl and monitor those who manipulate food safety and security. Restaurants, not all of them are able to between gluten and similar organisms. As you know, if a woman has gluten sensitivity, if she's a and eats food with gluten, she might have a miscarriage. A child might die or have complications. I believe that our collaboration is strategic, and with all the unified efforts, we will be able to make a change on the ground. The number of our patients is 250,000 in Jordan, and currently we are targeting pharmacists, doctors, universities. We provide training. We also provide trainings at hospitals, and we believe our partnership with you is going to be a strategic partnership. And one more time, we thank you for inviting us to this symposium and considered us as a main player. Thank you.
Said, you know, it's you are a non-for-profit organization. You're not receiving any funds. It's great to see the level of collaboration and partnership that you're getting from the Arab Army, from the military laboratories, but also with all the other partners, both government and non-government. In fact, actually, I would like to seize this as an opportunity. If there aren't any other comments, so there is maybe one comment or question. Yeah, please proceed. Is there a microphone that is close by that we can get you? Thank you.
هلا بس يعطيكم العافيه، بس انا عندي سؤال لمهندسه اروى. هلا انتم عم تعتبروا انه gluten free product is less than 10 ppm or less than 20 ppm? Because
هل تعتبرون المنتجات الغذائية الخالية من الجلوتين 20 دي بي ام أم 50 دي بي ام؟ بالنسبة لي عندما أجي اختبار لأي منتج غذائي فأنا بعمل كوانتيفاي ل 20 دي بي ام، ولكن أنا يعني rest assured that you are abiding by the adopted regulations. The less than 20 dBm is considered to be safe. Are you talking about the intervention value? Are you talking about the intervention value? The 20 dBm, where it came from? The studies had proved that the 20 dBm, the celiac patients will start to show symptoms. And the second reason this number was chosen is it's the minimum concentration that was targeted in all the testing techniques, whether the rapid method or the testing kit or the CBR. However, I would like to confirm that 20 dBm is considered to be the threshold. And there are and the Jordan Association for Specifications and Meteorology, they don't accept a concentration less than that. There are different types of allergies, such as peanuts, sesames, etc. What is we need to be provided with a threshold of allergens, and we would like you to share with us the general threshold for the total list of allergens. Because out of experience in my work, we usually read the Japanese standards. It's evidence-based research, because the same for agriculture as well. So sometimes I rely on the detection level of the kit I use. However, the detection limit is not necessary to a VPM. Is it approved or not? Some researchers say 10, other researchers say 8, or AOAC can share with us the most common thresholds of allergens. For instance, if you want to arrange or classify the intervention level for food products, we need them to be approved.
So here maybe if I may intervene. Clearly for gluten, there is an international standard and also national regulations that generally vary between 20 and 10. Most of them are 20 ppm because most of them are actually aligned with Codex. For allergens, there is no regulatory threshold related to the presence of allergens. And therefore, I guess the responsibility of laboratories is to report any level that is above the limit of quantification. Generally, if you want to do quantitative level, so you report any level above the limit of quantification. After that, it's basically based on a risk assessment that needs to be made. Maybe Codex will come up with some guidance, and the adoption now not of regulatory thresholds, but of health threshold. We have to make the difference between a health threshold, which is essentially the level above which there is a reaction, and a health-based guidance value, and a level that is occurring in food. So before we get to a level that is occurring in food, it will take some time. I can tell you the only jurisdiction that I'm aware of that has been using a regulatory level for allergens is Switzerland, with 10 parts per million. Okay. 10 ppm. 10 ppm.
Yes, that's true.
That's it. That's the only jurisdiction. But
there are some research that will tell you about the risk assessment or the intervention level, even if you found it. For me, we actually report the exact number, but sometimes المختبرات لها دور consultation مع their customers.
Sometimes the laboratories conduct consultations with the customer. So customer, they might say they want this or that. So we give the recommendation based on the thresholds.
There is a list essentially that was issued by the expert consultation.
I took them. I took the photo of
Those
them.
are the values. Those are the health-based guidance
This
values.
is the health-based.
Yes, there is nothing else.
Because they vary with something I already found before. That's why I'm asking about, because I took the a photo of the.
It's really a complex process to move from a health-based guidance value to an actual number in ppm. When I say it's a complex process, you need to do an exposure assessment, a risk assessment to identify how much of the population you want to protect,
population and do them.
It's in fact, actually, we're going to come to that maybe as a conclusion of this discussion, at least for gluten. So,
okay, thank you very much.
I think there was another question here.
أنا عندي تجربة بس عندي سؤالين.
I have two questions. One for the participants regarding test ELISA. We have problems with it in the laboratory. And the other thing, Ilham talked about it, labeling and mislabeling, and the use of a gluten. The manufacturers always say use any opportunity to write that their products are gluten-free. So they like to do that. It attracts customers. For instance, once I read the word halal or halal on a wine, how a wine can be halal. Do you think that by labeling halal would it increase the sales? So we need to fix the labeling system. So frankly speaking, in general, in the Arab world, and especially in Lebanon, there had been some initiatives to produce gluten-free products. However, the trader during the shipment process of corn flour, there might be cross-contamination from the vessel to the plant until it reaches the end user. We work on AI by using a QR code with a program connected with database of the laboratories. We have made a small initiative on honey for anyone who collects honey. In the laboratory, you would have a QR ceiling that is connected with the breeder or with the hive farm. I'm thinking loudly. Do you think that the gluten issue in coordination with the competent authorities, in order to have a connectivity starting from the importer up to the end user, in order to exert a pressure on the manufacturers to prevent them from labeling their processed food as gluten-free? And this can be done in collaboration with any entity of your choice. And with ELISA, we are working on a database with some partners. It's a bit complicated at the moment. However, my question for everybody, are you suffering with ELISA like us? Because I have a problem with accuracy, credibility and sensitivity. In 20% of the cases, I found that that the manufacturers who label their products with gluten-free, they put it on eggs or not cornstarch, as of course. Can you share your experience in dealing with ELISA, especially in importing food products? Can you share your experience?
There is two persons. ELISA result give false. يعني هاي يعني مسجل.
But again, I think I should maybe ask Stefan, did you hear the question that was posed? Would you like to take part of that, or?
No, sorry, I didn't hear, or could not
Yes.
understand
So
partially.
the general question was the following, is that one of our contributors here is expressing that he's facing some issues with the performance of some ELISA tests working on gluten or destined to gluten testing, particularly the fact that, again, I mean, the food matrices are complex matrices, and he's facing issues of, I think, repeatability, reproducibility, sensitivity, even actually, and accuracy. So would you be able to comment on that?
Yes, if you look at the study data that I showed you, only a part of it, if you look into the publication, you will see perfect data for different types of matrices regarding reproducibility from lab to lab, inside a lab, repeatability, accuracy. So, of course, you have to expect a kind of variation, but it is typically accepted not only for gluten, also for the allergens by the AOAC to be in a range of 70 to 130% recovery, because everybody knows that looking at different food matrices, you have different impacts on recovery. So some may show 70% recovery with the same analyte, and others may show 120% recovery. This is something that we have to expect. However, if you look, and therefore we did this study, if you look at the data, you see from lab to lab, independent where, if it was in Australia, USA, and Europe, we got similar results. So therefore, the accuracy of the method is good. Regarding complex matrices, we have to validate each matrix before testing. We have chosen here a wide range in our study of different types, from fish to pastries to dairy products. Nevertheless, the variation inside food is so big, and there are so many different ingredients which may have an impact on recovery, that you have to be careful regarding using whatever method you like to use. This is something which is true for every method that you choose. You have to carefully validate your metrics.
Thank you so much for a clarification. I will try just to say what is really the challenging we are facing in all kinds of lab testing, but I will focus on gluten test especially. When you say that the maximum acceptable limit is 20, our challenging here, when I get, for example, 23 or 15 or. So here, the stability of the sample or the kit, sorry, or the accuracy or the repeatability or the reproducibility here, I cannot judge for the customer here. Because if I have 5% error, positive or negative, adding or removing, so here it's really so hard to say it is absence or not. So this is really what we are facing in ELISA. When I repeat the test, maybe the error will be different between two lab technicians. So that's why I'm asking what is, yani, yes.
So I can give you actually my experience or the experience of our lab actually in this regard. And it's pretty much actually similar to what Stefan was mentioning. First of all, you need to make sure that with any new matrix that you are using, your test has been ascertained for its performance on that matrix, including an attempt actually to determine what is the percent recovery. And on the basis of that, you can also give a bracket of variance of your result. And that has to be accounted for in the way the regulatory decision is made. Because at the end of the day, as you say, you're the laboratory. So you report the result with the variance of the result, if you have the variance of the result. After that, it is up to the regulatory authority to use that information and be able then to make a decision. And the fluctuations related, or the potential fluctuations that are related to the performance of the method, should be an element of the decision making. I mean, this is not unheard of. In the world of nutrition, for example, for declaration of nutrients, you can have 50 and 150%, sometimes 200% accepted as part of the reporting in order to make a regulatory decision. So it's not, I would say, in the world of regulatory decisions, it's not necessarily new. The main element, though, is we need to know. Yani we need to know how the method performs. And that's, I think, the key element. To know how the method performs, there are two ways. Either the method has been ascertained through reports, through, for example, what we would like to do as part of the Arab AOAC work, which is essentially to document the performance of methods on matrices that are different from the ones where publications have done such reporting. So that's one of the ways. Or as part of your exercise in your own lab. Yani in our own regulatory laboratories, whenever there is, I remember we used to do that in Canada, whenever I have a new matrix that is out there, if it's not listed in the scope and I would like to check on doing that, I will do some trials on the method using verification samples and so on, just to be able to know how does the method perform. And then after that, I will use that information for the unknowns, because that's the only way we can do it. Dr. Rawan, you would like to?
Dr. Sam and Dr. Smith, you pretty covered the issue. I have no more to add, but regarding our analysts and our results, we do not have any concerns regarding this issue. Taking into consideration applying the GLP and the good analysis practices, validation and so on. So we offer to give you a technical advice or something like this regarding to this issue.
Thank you for that. Yes.
آخر تعليق لسؤالك الأول اللي هو مشكلة الليبلينج حتى
One last comment regarding the labeling problem. It's not enough to have standardized specifications or testing. Sometimes there is a gluten, invisible gluten that might be in one of the components. Sometimes even the spices might have a problem with the gluten. And thank you very much. Thank you one more time. I believe that we are close to lunchtime, so I try to recap before wrapping this session, and also to identify the future goals. There is a general agreement to exert more efforts in standardizing the analytical methods utilized, whether for allergens or in gluten testing. I believe the gluten issue has more attention, and there is an opportunity for more collaboration in the Arab region with the international bodies and organizations. And here maybe we can put ourselves in a group challenge, which is to work together to work on documenting the performance of the analytical methods utilized in gluten testing. And after the documentation, maybe we can standardize these analytical methods their performance in the targeted food products. And here, maybe we need the partnership with the CDI Society in Jordan and and other bodies and entities in the Arab region to identify our needs. What are the food products or agricultural methods that can be targeted to study the performance of the analytical methods? And hopefully maybe we can generate data to give us an idea about the contamination, to prove that gluten is present in these products. And this will help us develop a risk assessment tool in order to enable us to describe the degree or the level of risk for consumers in an attempt to avoid gluten by customers or citizens. And also and to study whether the threshold of a 20 dB of a gluten, hopefully we will be able to work on organizing or starting a joint project. Of course, one hand cannot club alone. We will seek every possible support or partnership and in-kind contributions, and also to provide funding. So here we open the door for any one who would like to participate and get engaged in this group challenge in an effort to overcome all the challenges and barriers in collaboration with the AOAC International, the Arab section. And hopefully in the next meeting, we will be able to provide a report that indicates the advancement of our work. Good luck for all of us. Stefan, we will ask the support of some companies such as the Arab Farmers Organization and other competent agencies. Thank you very much for participating in this session and the discussion. And now we would like to invite you to join us for lunch, and try to join us again at 2:15 p.m. Jordan time. Enjoy your lunch. Bon appétit.
Hello, can you hear me? Check. Yes, we can hear you. Hello, thank you for an introduction and for the invitation to speak at the second symposium of the AOAC Arab section about general aspects of involvement of AOAC International in Codex proceedings. It's a pleasure to be here, and I would like to say hello to everyone. AOAC International is an international organization with 15 regional sections, which are listed on this slide. One of the important missions of the sections is to gather people during regional organized annual meetings, such as your symposium. And AOAC sections also provide an important insight into regional analytical needs and enable collaboration on global alignment of trusted analytical solutions, which is the vision of AOAC International. Our mission is to advance food safety and product integrity through standards, validated test methods, and laboratory quality programs. And this vision and mission are also well aligned with the Codex Alimentarius, which is why AOAC has been actively involved with Codex. So what is the Codex Alimentarius? The Codex Alimentarius, or Food Code, is a collection of standards, guidelines, and codes of practice adopted by the Codex Alimentarius Commission. The Commission, also known as CAC or CAC, is the central part of the joint FAO-WHO Food Standards Program, and was established by FAO by the Food and Agriculture Organization of the United Nations and WHO, the World Health Organization, to protect consumer health and to promote fair practices in food trade. The Codex Alimentarius Commission held its first meeting in 1963, so it celebrated 60 years last year. And during those 60 years, Codex has developed hundreds of internationally recognized standards, guidelines, and codes of practice. It has defined thousands of permitted levels of additives, contaminants, and chemical residues in food, including maximum residue limits for pesticides and wet drug residues. As I mentioned, the Codex Alimentarius is a collection of standards, guidelines, and codes of practice, which are adopted by the Codex Alimentarius Commission. Those are so-called Codex core texts, which help ensure that food is safe and can be traded by preventing and helping to resolve potential trade disputes. Codex texts are not mandatory, but governments frequently use them as the basis for national legislation. There are currently 237 standards, 87 guidelines, and 57 codes of practice in the Codex. I have listed here just a few examples, including a very important Codex Standards 234 on recommended methods of analysis and sampling, which I will discuss in more detail later on because it contains a lot of AOAC official methods. The acronyms such as CCMAS or CCFL or CCNFSDU, etc., are various Codex committees that are responsible for those standards, guidelines, or codes of practices, which cover many different topics, from general topics such as general standards or principles to very specific ones such as standards for specific commodities, which is the, for example, the last standard example, which is listed here. It is a recently adopted standard for berry fruits. And Codex standards can take several years to develop. To The process has 8 steps, which are shown here in the slide. It starts with national government or a Codex committee making a proposal for a standard to be developed. They then prepare a discussion paper that outlines what the proposed standard is expected to achieve, and then a project document that indicates the time frame for the work and also its relative priority. And as I mentioned, standards can take several years to develop. Once they are adopted by the Commission, a Codex standards is added to the Codex Alimentarius. If you're interested, you can find a lot of useful details about Codex rules and procedures in the procedure manual of the Codex Alimentarius Commission, which can be found on the Codex website. It also includes guidelines for the operation of Codex committees. And there are 3 types of Codex committees: general subject committees, commodity committees, and also FAO coordinating committees. You may also know them as horizontal, vertical, and regional, and I will explain why. There are 6 Codex regions, which are represented by a joint FAO regional coordinating committees. Each of these regional committees is responsible for defining the problems and needs of the region concerning food standards and food control. The regions can recommend to the Commission the development of global standards, but can also develop regional standards for food products traded exclusively or almost exclusively within that particular region. These regional committees play a general coordinating role for the region and also work to promote the acceptance of Codex standards and maximum limits for residues and residues by member countries. In addition, they should also promote coordination of standard development activities by international governmental and non-governmental organizations within the region, which includes development of AOAC standards and official methods. And I believe that AOAC sections can play a significant role in this effort and collaborate with Codex regional committees. The Codex commodity committees, sometimes called as vertical, prepare standards for specific commodities. Codex commodity standards define the physical and chemical characteristics of nearly 200 traded products. And there are currently only 4 active commodity committees. There used to be many more in the past. The 4 active committees cover fish and fishery products, fresh fruits and vegetables, fats and oils, and spices and culinary herbs. The general subject committees, sometimes referred as horizontal, because they establish standards and guidelines and codes of practice applicable to all foods horizontally, to all products and product categories. And they deal with contaminants in foods, food additives, food hygiene, food inspection and certification, food labeling, general principles, methods of analysis and sampling, nutrition and foods for special data uses, pesticide residues, and also wet drug residues. And the most important Codex committee for AOAC's work is CCMAS, which is the Codex Committee on Methods of Analysis and Sampling. And in the recent years, AOAC International has been also very actively involved with CCNFSDU, which is the Codex Committee on Nutrition and Foods for Special Data Uses. And I will explain why very shortly. But first, how can an organization like AOAC International get involved in Codex? AOAC is not a member. Codex members are countries. Currently, the Codex Alimentarius Commission has 189 Codex members made up of 188 member countries and 1 member organization, which is the European Union, the EU. In addition to members, there are Codex observers. Currently, 240 organizations are Codex observers, which can participate in the Codex process except for voting. And AOAC International is one of the Codex observers. The key role of AOAC in Codex is to provide AOAC official methods, which are included in Codex Standards 234, on recommended methods of analysis and sampling. There are many AOAC methods included in this Codex standard. In fact, there are currently 384 AOAC method references listed in this standard. AOAC also develops standards and official methods for existing Codex provisions and submits or recommends AOAC official methods for endorsement by Codex committees. We play an important advisory role on Codex provisions, methods, requirements, and guidelines, procedures, and so on, and also participate in Codex committee meetings and working groups, and also participate in CCMAS interagency meeting, or IM, which I will talk about later. As I mentioned, there are currently 384 AOAC method references listed in the Codex standard 234 on recommended methods of analysis and sampling. And this standard is amended every year. And here's an example table from the standard, just one page showing methods for fish and fishery product commodities and provisions such as histamine or mercury and so on. And you can see a lot of AOAC methods listed in the method column on this page. Some of them are quite old, developed way back in the last century, but still important for Codex provisions. But there are also some very new methods which were recently adopted by Codex, especially methods for the analysis of various nutrients in infant formula, which were developed by SPIFAN, which is the AOAC stakeholder program on infant formula and adult nutritionals. And there are 26 of the SPIFAN AOAC official methods included in the Codex Standards 234. They are listed in this table, and you can see that many of them have become also ISO or ISO IDF standards. And in fact, the collaboration between AOAC and ISO and IDF started thanks to our involvement with Codex, and it continues. The SPIFAN methods that have been adopted by Codex were first brought to CCNF SDU, which endorsed recommendation of these methods and proposed their inclusion into Codex Standards 234 to CCMAS. And CCMAS then endorsed their inclusion, which was adopted by the Codex Alimentarius Commission. When it comes to the Codex Committee on Nutrition and Foods for Special Data Uses, or CCNF SDU, the aim of this committee is to study specific nutritional problems, which are assigned to it by the Commission, and to advise the Commission on general nutritional issues. It drafts general provisions concerning the nutritional aspects of all foods. It is a horizontal Codex committee. It develops standards, guidelines, or related texts for foods for special data uses. This includes infant formula, and also considers and endorses provisions on nutritional aspects which are proposed for inclusion into Codex standards, guidelines, and related texts. And as I mentioned, AOAC has been actively involved with CCNF SDU in the recent years when it comes to the SPIFAN methods for nutrients in infant formula. And many of these methods have been also validated in foods for special medical purposes, or SMPS, which is our next focus. But in addition to that, we are also planning to discuss methods for dietary fiber at the upcoming CCNF SDU meeting later this year. AOAC has been also actively involved in the discussions related to methods of analysis to measure sweetness, which is a very complex topic. And we are also involved in the work on optional ingredients in the infant formula standard, because some of the more recent SPIFAN methods were developed and validated for optional ingredients and couldn't be adopted by Codex due to the lack of provisions. And one nice highlight related to CCNF SDU is that last year, the AOAC delegation representatives, Daryl Sullivan and Eric Konings, were invited to provide educational presentations at the CCNF SDU meeting on the SPIFAN program, on methods and also related analytical aspects. You can see some pictures from from that meeting and their presentations on this slide. As I mentioned, CCMAS, or Codex Committee on Methods of Analysis and Sampling, is the most important Codex committee for AOAC's involvement in Codex. CCMAS objective is to define the criteria appropriate to Codex methods of analysis and sampling, is to serve as a coordinating body for Codex with other international groups, such as AOAC International, is to specify reference methods of analysis and sampling appropriate to Codex standards, and consider or amend, endorse methods of analysis and sampling proposed by Codex community committees, Codex committees, such as also CCF and SDU, except that methods of analysis and sampling for residues of pesticides and bedrock residues, the assessment of microbiological quality and safety in food, and the assessment of specifications for food additives. These do not fall within the terms of reference of CCMAS. The objective of CCMAS is also to elaborate sampling plans and procedures and consider specific sampling and analysis problems, and to define procedures, protocols, guidelines, and related texts for the assessment of food laboratory proficiency. So CCMAS is really very relevant committee for AOAC, as you can see from all these different tasks that CCMAS has in its scope. Examples of recent AOAC's involvement with CCMAS are certainly all the endorsements of the AOAC's PIFN methods for nutrients and infant formula. As I mentioned, 26 methods have been included in the Codex Standards 234 since 2016. But we are also getting involved in other areas, such as food allergens, where we have AOAC Gluten and Food Allergens, or GFA program. This program has been focused on development of validation guidelines, which were just approved, so we could include them as a reference in our comments on the electronic working group discussion paper on methods of analysis for precautionary allergen labeling. This is electronic working group within CCMAS. These comments were provided by Dr. Melanie Downs. She's the chair of the AOAC GFA's working group on food allergens. And Melanie is going to join the AOAC Codex delegation during the next week's meeting of CCMAS in Budapest. And we have been involved on other CCMAS electronic working groups listed here on the slide. The last one on the discussion paper on listing type 4 methods in Codex Standard 234, when a type 1 method is listed for the same commodity and provision, is a very important topic for AOAC and other SDOs. So we collaborated on our comments with other organizations that are involved in so-called interagency meeting, or IM. The interagency meeting, or IM, is an informal group of standard development organizations, or SDOs, which are associated with CCMAS. IM meets before the CCMAS meeting or as needed. Nice highlight is that AOAC International, Daryl Sullivan, has recently assumed IM's vice chair role. The interagency meeting promotes coordination and cooperation among SDOs, and it addresses problems that require international collaboration. It assists CCMAS in advisory capacity and collaborates on position documents. Some examples of recent Codex Room documents, or CRDs, which were led by AOAC, include the already mentioned comments on the electronic working group findings of concerning the adoption of type 4 methods in the Codex Standard 234 when a type 1 method is listed for the same commodity and provision. These were submitted to this year's CCMAS meeting. Or an example of a previously developed position that CCMAS should serve as the nodal committee for all analytical methods with Codex provisions. I mentioned when I was discussing the exceptions where CCMAS currently does not have a role. So AOAC and other SDOs propose that CCMAS would be the nodal committee for all analytical methods within Codex. And with that, I want to highlight our involvement in this year's meetings. As I mentioned, there's going to be a CCMAS meeting next week in Budapest, in Hungary. And I believe that the next speaker is going to highlight some of the important topics which will be discussed during this meeting, so I didn't go into these details. We are also going to be present at CCNFSDU meeting later this year in October in Dresden, in Germany. And with that, I would like to thank people who've been heavily involved with SPIFN method endorsements and related Codex activities. Daryl Sullivan from Eurofin Scientific, Eric Konings from Nestle, and Dustin Starkey from Abbott Nutrition. And this picture is from last year's CCMAS meeting. And I also want to thank my colleague, Deborah McKenzie, who is my deputy and AOAC Chief Standards Officer for all her work on AOAC standards, including the standards which has been adopted by Codex. For those of you interested in AOAC activities, not just about Codex, please follow us on LinkedIn, and you can also subscribe to weekly email updates. Here's a QR code for how to subscribe to Spectrum, which comes into your email every Friday. And with that, I would like to thank you for your attention and be happy to take any questions. And again, here's the QR code for Spectrum if you're interested to get updates from AOAC International weekly. So thank you so much, and I'll be happy to take any questions.
شكرا كاترينا.
Thank you, Katrina. We receive all questions at the end of the session.
Thank you. Thank you.
Thank you, Kat. طبعا هيئة الدستور الغذائي كما يعرف
Of course, the Food Constitution Commission or Codex Commission is an international food standard setting body, the central part of Joint Food Standards Program. The commission is comprises 189 members, including 188 member states and one member organization. The commission or Codex developed international standards for safety and quality products to enhance food safety, which increase and increasing its nutritional value to consumers around the world. In addition to the technical committee, the Codex is the umbrella of committees. The first one is the which is in charge of all issues related to the execution of the food specifications, and the committee meets annually between Rome and Kiev. And also there is the executive committee which can submit suggestions and recommendations to the committee regarding the general tendency and the program, under which the coordination committee for Asia, coordination committee for Europe, and the other one for Latin America and the Caribbean, and committee for America, and regarding the technical committee under the Codex, products, they are divided into the the general committee and the and commodities committee. As for the commodity committee, there are four active committees. There's the fishery products, the fruits, vegetables, spices, and for the general subject committees, the 10 for the contaminant food and food additives and the veterinarian committee, the general principles committee, the food labeling committee, and the methods of analysis and the sample committee, the nutrition and food for special uses committee, exercise committee, and the residues of veterinary drugs in food and the food hygiene committee. As for the action and sampling committee, which is held in Hungary, 15 documents had been issued, and you can review these documents by visiting the website of Codex for free. For instance, the specification of Codex number 1704 regarding the ISO standard specification 204 that update was in 2020 by review standard. You would notice that it covers a number of sectors, and these tables you will find all the required inspections and tests for each product, and some products require several tests in addition to all the issued resolutions of the AOAC. To talk more about the analysis committee and sampling, its meetings are conducted periodically, and the third meeting is going to be conducted in next this month. In view is of the meeting, over a number of important things are going to be discussed. Most important, after the meeting, agenda is adopted. The analysis methods are going to be endorsed in addition to the and the plan of sampling is going to be created and opted, and the discussion of work criteria and decisions regarding the processed food and fruit and vegetables products, and in addition to the foundation, the third Codex Standard 234. Learn more information about the agenda by reviewing or visiting the Codex website. Based according to time constraints, it's impossible or not possible to analyze all or to discuss all the topics included in the agenda. They're going to deliver or present example. The fourth meeting, the sampling process and analysis methods of fish products. During the meeting, it was discussed to get rid of all the contradictions and to amend the procedures and steps in a way that fits the purpose, and review all the provisions which were developed codex regarding fish and fish products, and also discuss all the approvals and to endorse also to check whether the enlisted methods of certification fish and fish products to be part of the food review 234 and to see if there are any listed. So verify if the methods listed in the CCFPP standards are included in the CX 2034 and to verify whether the methods listed are. So the committee of sampling and analysis are supposed to review the appendix number 1 and to consider the proposed recommendations and suggest relevant methods where placements are recommended, and also to endorse the recommended revisions to CX 234 and third to recommend the CCFP to know of the revisions and to consider. And as a final note, I'd like to recommend to communicate with review all the documents and to inform all committees with the feedback in a way that's positively reflected on our work. Thank you very much for your listening, and we are ready to take questions during the Q&A session. Any questions? We usually exchange correspondence with the Ministry of Agriculture and the Ministry of any questions regarding the presentation or the second presentation. We are ready to take your questions. I believe there are no questions. Go ahead. Kindly use the microphone to speak.
يعطيك العافية. الله يعافيكي. هو مش سؤال أكثر من إنه
Good afternoon. It's intervention, not a call. We'd like to thank the Jordan Standards and Meteorology Association. This is first there. And I'd like to comment also on gluten-free product. It's my favorite topic. I believe that we will conduct a review of the documentation on this topic. Thank you very much. It was a very interesting presentation. Thank you very much. Do you have any questions?
Thank you, Dr. Katrina.
Thank you, Dr. Katrina.
Thank you. Bye-bye.
No questions. Thank you very much, Jireen, Basma, and Dr. Katrina for the fruitful presentation.
Between all these international standardization organizations and how AOAC is implicated in the Codex Alimentarius work. And as you mentioned, Dr. Kate, AOAC has different implications and involvement in several committees. Of course, mainly the CCMAS committee, the Codex Committee on Methods of Analysis and Sampling, but other Codex committee. So Dr. Kate, if you wish to add some closing remarks, the floor is yours.
Well, thank you very much. And I hope that we can get engaged more. And I also, as I mentioned, to be nice to engage the sections, right, on the regional issues. So I look forward to the collaboration. And I also want to invite you to the annual meeting. If some of you can come, you can see in my background, Baltimore, where we have the annual meeting in the end of August. And I want to wish you a very nice rest of your day and successful end of your symposium. Thank you.
Thank you so much. So a round of applause. حضورنا إلى نهاية المؤتمر.
Distinguished guests, we have come to the end of this symposium. Allow me to provide a summary of the topics been discussed. So this symposium had included five sessions. The first session entitled the test program and the AOAC national and also the current effort collaboration with organizations inside Hashemite Jordan Arab region, collaboration with the AOAC and with the support and the encouragement of the AOAC as well. So through this session, I would like to remind you that remind you of the importance of proficiency tests in the Arab region, which is encouraging the Arabian project, the Arab Proficiency Testing Program, with the support of all competent Arab organization entities and also AOAC International. And since the collaborators with us, we would like you to complete the survey. You will extend the duration another month. We will urge you and encourage you to create it. Would like you to identify priorities regarding proficiency testing. Regarding the programs we had already developed and adopted AOAC International, and we have a priority to launch the work reading the program of go ahead, Dr. Ruba.
If you put on the microphone, please.
On the microphone. All your opinions are important. Idea is that directors of laboratories within your institutions or entities to complete the survey, and we would like the competent personnel to complete the survey. I believe the technical people are the ones who should be completing the yes, the result will be shared with AOAC International, and accordingly, priority will be identified. We are waiting for your responses. We need big number of answers in order to have scientifically adapted information. And regarding the next session, it dealt with the analysis of pesticide residues, and he had a number. We listened to our presentations that are highly scientific, high level scientific, important points that had discussed that is to facilitate collaboration between Arab bodies and laboratories and the international bodies and organizations. And I would like to thank you for the active participation in that session and the Arab section of the AOAC International, and in an attempt to get a database, a scientific database
في المنطقة العربية لتسهيل التواصل فيما بينها. بما يخص انطلقنا اليوم الجلسة الثالثة بما يخص
and in third session regarding food fraud, which was very important topic. I noticed how important it was through the discussions that took place among the participants and analysts, and also we discussed the non-targeted approaches and food analysis in the region. And here, allow me to welcome His Excellency Emir الصقر سعد الصقر. The head of the Arab section of the AOAC International. You are most welcome, sir. With regards to the fourth session that was conducted later, we discussed the gluten allergens, and you had allergens and the gluten allergies. You had active participation during the session and highly technical, and a number of lessons learned are shared among the participants and pointers regarding the analytical method. Here, we looked at the second initiative which requests to share the adopted and utilized analytical methods in the region regarding gluten testing. And here, I would like to remind you to share the analytical methods that are adopted and used in the laboratories, especially to analyze and test the Arabian food product, because in a program of standardization of food allergens, hopefully we will be reached a unified or standardized testing analytic methods, gluten test. The last session was on codex committee analysis methods and sampling in cooperation with AOAC International as an observer. And also, we listened to the different activities conducted by the committee. These committees like to receive your feedback. Thank you very much for being here with us, Professor. Would you like to say a word on the mic, please? I would hope that the GMO will have a special session in the symposium or meeting, because for some problem with the GMO, it's an problem is only for it's the whole Arab world. Thank you very much for your opinion. If you have any suggestions for the next symposium, please hesitate. Thank you very much. Now we move to the final session, official session of today's meeting.
بسم الله الرحمن الرحيم
In
والصلاة
the name of Allah, the most gracious, most merciful, and peace be upon our Prophet Muhammad. Your Excellency, the Director of the Military Laboratories for Quality Control, Khadr al-Dawiyya. Your Excellency, Director of JISMA, Dr. Asagir Asri, Director of the G-Force, Dr. Samuel Godefroy. Peace be upon all of you. We welcome you today in the closing ceremony of the second symposium of the AOAC Arab Action, which was organized by the Jordan Force, the Arab Army, in collaboration with the G4 and the AOAC. Your Excellency, the symposium has included and covered the different presentations on proficiency testing, in addition to presentations on pesticide-induced food fraud and food allergens and gluten and their method of analysis. Representatives came from different organizations working in laboratory analytics. The success of the symposium is considered a cornerstone to continue our efforts to organize future symposium. I highly appreciate efforts exerted by the experts who participated, input and recommendations. Your Excellency, distinguished guests, now the Director of G-Force, Dr. Sam Kotov, will take the floor for the closing remarks. In the name of Allah, most gracious, most merciful, Karan Khadr, Director of Military Laboratory for Quality Management, Your Excellency Engineer Saqr Al-Saqr, distinguished guests, members of the Arab sector of the AOAC. At the end of the symposium, I would like to express my deepest great thanks and gratitude to everyone who contributed to the success of the symposium. On top of where our senior management and leadership of the Jordan Armed Forces, the Arab Army in collaboration with the AOC, the National Arab Section, all the speakers as well from the Hashim Kingdom of Jordan, specifically GFDA, GESCO, the accreditation unit, in addition to all partners from private and public sector. Thank you very much for hosting us in Jordan and all the efforts that you exert to guarantee the success of this symposium. The presence of His Excellency, the Director General of Adel Al-Saqr expresses the amount of importance that the organization places on food safety, agricultural products, and industrial sector, and it also highlights the role analytical procedures play in standardization and in improvement of assessments in the Arab countries to enhance intertrade between Arab countries. We hope that our symposium has contributed in supporting the Arab analytical community and in exchanging activities and information between experts of agricultural and food sector, and we hope that it has supported our future vision in standardizing analytical procedures in food control and conformity tests. Again, we would like to extend our greatest and deepest gratitudes to everyone who organized the symposium, on top of which is the Jordan Armed Forces, the Arab Army, in addition to the Army Farm supporting AOC International, in addition to Water Corporation, and we would like to thank our colleagues from G-Force, in addition to the chairman of the Arab sector of AOC International, Dr. Amman Kassouf. Hopefully, we will meet again soon in another country of Arab region, and hope that we will take advantage of the time between the two meetings to implement a number of the projects that we have discussed and agreed on. Peace be upon all of you. And now, Excellency Director of the Military Laboratories for Quality Control, Colonel Staff Khadr Ad-Dawoodiya for his closing remarks. Peace be upon all of you. In the name of Allah, most gracious, the most merciful, and peace be upon Prophet Muhammad. Your Excellency, Director General of the Standardization and Standardization Organization, the engineers, we are very happy that you joined today. We were hoping that you could attend yesterday's event, but we understand that you had other engagements and are very happy that you are here to attend the closing ceremony. Your Excellency, Director of G-Force and distinguished guests. In the beginning, allow me to express my gratitude for participating in the closing ceremony of the second symposium of the AOAC International Arab Section, which was under the patronage of the Jordan Forces Arab Army. I would like to thank everyone who contributed to the success of this very important event. We also apologize for the minor technical mistakes in the invitations that we addressed, and we managed to reset the invitations. Again, there were some spelling mistakes, email addresses, and we addressed the issue. And we also appreciate the RP of some of the organizations who could not make it due to other engagement of theirs. We have gathered in front of this symposium for 2 days, and we exchanged our expertise and the knowledge of ethical procedures and latest technology used. Everything was presented by experts and highly qualified individuals. And to be honest, there were several things that were of interest to me. I'm sure I was following most of the presentations. We all know that we speak the same language. The language pertaining to laboratory tests and accuracy of these tests will all drive us towards safety. Food safety at the level of at the national level. If we achieve it at the national level at Hashemite Kingdom of Jordan, it would definitely reflect at the regional level and onto other Arab countries. This is why everything that was presented on all topics were very important. And I would like to choose one topic because we are limited and we cannot delve into the details of all the topics that have been discussed. I personally was interested in the gluten topic and the panel discussion, which especially panelist Al-Qaisi, who has indicated that there's a large category of the community who are affected by the gluten sensitivity, and that there is a large scale of traders who unfortunately do not pay attention to this affected category. During this symposium, I noticed that there are several people participating, including the ones participating online. I believe that all of us should make more studies on this category of gluten sensitive individuals. To avoid the people suffering from this condition and to make sure that we are helping them. They are members of our families and our friends. We have to work hand in hand to address this issue related to gluten sensitivity. laboratory in general. We have gathered here during the symposium. We have discussed all the topics related to our work, and I was very delighted to note that even during break, people were discussing technical topics and they exchanged their business cards. This is the most important. Networking is very essential. I have personally met directors of laboratories I've never met in them before, so this is a very good chance to meet them in person, which definitely broadens the aspects of collaboration, allows us to brainstorm and hopefully achieve implementation of several projects. I will not speak much longer since this is a closing ceremony, and I know that all of us are longing to go back home. I would just like to extend my thanks and gratitude one more time to everyone for their participation. Hopefully, we'll meet in future events and symposiums. And may God protect our beloved Jordan under the leadership of His Majesty King Abdullah II, the senior leader of the Jordan Armed Forces and the Arab Army. Thank you very much for your attention and for your presence. Distinguished guests, achieving the comprehensive objectives between the Joint Armed Forces, Arab Army, and G-Force in conducting studies and scientific research and innovating analytical tests and improving works. We will now sign an agreement for lending the CMS from G-Force, which is considered a great addition to the equipment of the military laboratories for quality control. And now we invite Colonel Khadr Al-Wadiya, His Excellency, the Director of AOAC, to the stage to sign the agreement.
والآن فليتفضل
And
سعادة
now I would like to invite His Excellency, the Director General of Dr. Engineer Adel Al-Saqar for his closing remarks. He is the Director General of AIDSMO. In the name of Allah, the most gracious, the most merciful, peace be upon Prophet Muhammad and his companions. Your Excellency, Staff Colonel Khadr Dawoodiya, Director General of Military Laboratories in the Jordanian Armed Forces, the Arab Army, Your Excellency Professor Samuel Joseph, Director General of the Force, distinguished guests, peace be upon all of you. Allow me in the beginning to extend on behalf of myself and AIDSMO, to extend our greatest thanks and gratitude to His Majesty King Abdullah II bin al-Hussein. May God protect him for his continuous efforts to support collaborative work between Arab countries, and for Jordan hosting a second symposium of the AOAC International Arab Section. I'm very delighted to be present with you in this very important event, and I would like to extend my thanks to Professor Samuel for allowing me and giving me the opportunity to be amongst you. And I hope that this was a successful event. I'm also very delighted to attend the memorandum of cooperation signing ceremony between the military laboratories and the force, hoping that it will bring success in the future. And I would like to thank AOAC, the Arab section in Morocco, where they work on developing analytical tests, and the organization encourages the efforts of its members in risk analysis and assessment, and enhance good practices to protect Arab consumers. Your Excellency, distinguished guests, there is no doubt that the memorandum of cooperation that has been signed today will open horizons towards creating cooperations between different laboratories, between GFROS and military laboratory in Jordan, in addition to providing training courses for experts. This will have a positive impact, especially in terms of food control and analytical methods control, and it will serve the Hashemite Kingdom of Jordan and other areas. Again, I would like to reiterate my thanks and gratitude, and I would like to congratulate you on the signing of this memorandum of cooperation and for the successful organizing of this symposium. Peace be upon all of you.
والآن فلتسمح
Now, allow me, Your Excellency, to start the ceremony for handing out plaques. His Excellency, Director of AIDSMO, Engineer الصقر عادل الصقر.
عادة رئيس المنظمة
Excellency, the head of G4, Professor Samuel Godefroy.
سعادة رئيس القسم العربي
His
لمنظمة
Excellency, the Chairman of the Arab Section for the AOAC, Dr. Amin Al-Qassouf.
فليتفضل الجميع
And
لصورة
now
تذكارية.
I would like to invite you to gather for a group photo.